On behalf of Anna Rose and myself, we're happy to welcome you to the first in-person T32 work in progress meeting since 2019, which was a while ago. And just as a little bit of sort of background, Anna Rose and Chris Pierce really got this translational training grant off the start exceptionally well. And after Chris left, I sort of stepped in. But really, Anna Rose has done all the work and continues to do all the work. But everyone, hold your sort of thumbs together. That's what we say in Germany. Wir drücken die Daumen. We're pressing our thumbs, which means crossing our fingers, because October 19th is when our T32 gets reviewed. So fingers crossed, we can continue another 6 years. Just happy to have everybody here. I'll introduce some of the specifics, but I think Anna Rose, if you want to say a few words.
I'll just say a quick hello and welcome. So a very quick hello and welcome to all the people who are logged on, who with this rain and this miserable set of temperatures are not physically here today, and also because of our COVID-19 regulations. But we're so grateful that since 3 years, we're finally some warm bodies in a room, and with some libations and some chocolate at the end of the afternoon. We're very grateful for it. As you know, our departments, pharmacology, psychiatry, place a great deal of emphasis on training. And so this grant that Julie refers to as something that Chris and I started is really a continuation of a long time T32 effort that was led by Dr. O'Brien for more than 2 decades. So he's sort of the paterfamilias of the Addiction Research Center at Penn that grew up inside the walls of the VA, finally split the walls and moved to 3900 Chestnut for about 26 years, and then has been at 3535 Market as its hub for the last 5 years for some of the clinical work. But we're really grateful to be able to have the continuation. And as Julie says, with good luck, the review in October will give us 5 more years beyond October, but 6 years from now. So we will keep our fingers crossed. So do we want to go ahead and you introduce your first person?
I think the first person is now the last person, because Daryl graduated, and unfortunately his actual last day was Friday, when he needs to come back for this afternoon. Daryl's an MD student with me, and he was very successful during his training on the T32 with 4 publications, his first authors, and one was submitted. And they'll tell us about bi-directional relationships between opioids and
All right, thanks everyone for coming today and for the great training I received through this grant, and it's nice to wrap it up this way. So I'd like to talk about the research I did during my thesis, which explored the relationship between opioids and sleep. And this is, okay. So it's important to still study opioids, because even though we've caught on that they are incredibly dangerous and addictive, and prescriptions have decreased in the last 10 years, the amount of opioid overdose deaths has actually increased substantially in the last decade. So it's really important to study all the biology relating to opioids. And the section I sought to focus on was a side effect of opioids and withdrawal, which is disrupted sleep. And it's been known for thousands of years that opioids and sleep have this relationship, because the Greek god of sleep, Hypnos, was often depicted sprinkling opium poppies down on the mortals that were having trouble sleeping. So it's been known in history that there's this relationship, but scientifically it's really been neglected until the last 10 years or so. But advances in optogenetics and chemogenetics combined with electroencephalography have shown that when you activate or inhibit these neurons that were previously canonically just reward-related neurons, it'll actually wake an animal up or put it to sleep, or vice versa, depending on whether they're weight-promoting or sleep-promoting neurons. So we found that these reward and sleep areas of the brain are actually way more intertwined than we previously thought. And then with our proximity to the sleep center here in TRL, we kind of took advantage of that collaboration to really dive into the fundamental neurobiology linking opioids and sleep. And even beyond weight promoting and sleep promoting neurons, there's circadian rhythmicity in these drug reward areas. So just as an example, this is in the nucleus accumbens of humans. And in unaffected controls that you see here on the left, they have this really striking circadian pattern where certain transcripts are expressed at certain times of the day more highly. This is about calculated from time of death compared to sunrise on that day. So the controls have this very nice circadian rhythmicity in the nucleus accumbens, but patients with opioid use disorder have this completely wiped out. So they have a total shifting and blocking of normal circadian rhythmicity in a region that's typically associated with drug reward. And beyond that, just looking at the patient population of opioid use disorder, the majority of opioid use disorder patients actually have a comorbid sleep disorder. 3/4 of them even complain of sleep issues. And those patients that do have sleep issues are at increased risk of relapse and have increased craving compared to opioid use disorder patients that don't have sleep issues. And then even 30% of opioid use disorder patients that try to stop say that sleep problems are a reason for relapse back to opioids. So including all these other side effects that we know from opioids, the poor sleep is actually pretty detrimental because it creates this paradoxical feed-forward mechanism where opioids cause poor sleep, which causes opioid use, et cetera. But we don't really know the biology behind this or where it starts or how to stop it. And then a further example of this is just on the left. These are patients dependent on prescription opioids, and they have higher scores on the Pittsburgh Sleep Quality Index and Insomnia Severity Index, both of which higher scores indicate worse sleep. And then on the right, the other way, looking at 60,000 subjects that reported sleep times, those that reported less than 6 hours of sleep per night are at an increased risk for all kinds of drug use, including opioid use. So again, this is more evidence of a feed-forward cycle where poor sleep influences drug use, which hinders sleep, et cetera. And so it's been my role to kind of dig a little bit deeper into this than just correlations from human populations and see if we can find a neurobiological mechanism explaining this. So looking at this bidirectional relationship, we kind of want to explore and close this circle. I had previously showed that chronic sleep disruption can alter voluntary morphine intake. So disrupted sleep can change opioid use. And for my presentation today, I'm going to focus on the reverse part of the circle. So how opioids can disrupt your sleep and specifically what neurons and cell types are responsible for this phenotype. So to start this, we used an oral morphine paradigm. So we put morphine in the drinking water for mice, and we masked it with saccharin, which can mask the bitter taste. So mice will drink enough to become dependent. As we see here, these mice are dependent on morphine as they show both significant spontaneous signs of withdrawal as well as somatic signs of withdrawal from naloxone that we precipitated withdrawal. So we developed this paradigm it's not stressful, it doesn't require a surgery or any injections, and then it's easy to kind of just put the morphine right in and take it right out to manipulate it. We used this paradigm to study sleep with electroencephalography. So we did an EEG surgery where we implant electrodes 1 millimeter from bregma and on the neck muscles and looked at a 24-hour period of sleep recordings with electroencephalography at baseline on the first day of morphine at this dose, and then 11 days into this dose when mice are dependent, and we call this the chronic morphine time point. So just to orient you, this top row is all baseline. So obviously at baseline before anyone has had morphine, there's no difference between the group that's randomized to control and the group that's randomized to morphine. On the first day of morphine, we start to get a phenotype where we see more wake and less non-REM sleep in the morphine group compared to the control group. But this is kind of a small and main effect, but nothing popped out in a post hoc test. But the real big phenotype we found is that chronic morphine significantly increases wake time during the dark cycle. So this is when mice are typically awake. So it's kind of an extra exacerbation of a normal circadian rhythm. So chronic morphine increases wakefulness, really sustained through the entire dark cycle period. And there's no phenotype during the light cycle when mice are typically sleeping more. So again, they have increased wake and decreased non-REM during the chronic morphine paradigm. So this is a nice phenotype that shows opioids can disrupt sleep in a specific circadian manner. And from here, we wanted to zero in on which neurons might be responsible for this phenotype. But having nowhere to start, we did a screen of the immediate early gene CFOS. So this is often used as a marker of neuronal activity because CFOS will be expressed after a stimulus with enough depolarization when we see these neurons firing. So we just looked at a number of brain regions that have been implicated in opioids or reward or sleep-wake or the combination, and did this combination of the same oral paradigm that we used to see the sleep phenotype, but then we did an injection of CFOS just because you need this systematic stimulus in order to induce CFOS expression. And what we found was that in the lateral hypothalamus and paraventricular nucleus of the thalamus, we saw a significant increase at the acute time point, but the only one that remained elevated was the paraventricular nucleus of the thalamus. And this was of interest to us because the PVT, as we call it, actually has these wake promoting neurons. So if you turn on these neurons, a mouse will go from asleep to awake. And if you inhibit these neurons, a mouse will sleep more. And then other groups have recently shown that injection of morphine alters the spike properties of these neurons, depending on whether the morphine's given in the light cycle or the dark cycle. So we have evidence from the literature and also from our paradigm that the PVT may be an important locus for the relationship between opioids and sleep. So we wanted to get kind of a genetic profile of these neurons. So we focused on mu opioid receptor expressing neurons because that's where morphine binds in the brain. So we took the mu opioid receptor expressing neurons in the paraventricular nucleus of the thalamus, and to isolate and sequence these, we employed translating ribosome affinity purification. So we have this mouse with genetically expressed an EGFP L10A fusion protein. So the L10A is a subunit of this ribosome. And then you can do an immunoprecipitation with a GFP antibody to pull down just the RNA from neurons that express, in our case, Cre recombinase in the mu opioid receptor expressing neurons. So we're getting RNA that's translating at the ribosome just in mu opioid receptor expressing neurons. And we did RNA sequencing on this specific cell type in this brain region that was of interest to our sleep phenotype. And basically this overall pattern shows that 11 days of morphine in the drinking water, the same paradigm we used to disrupt sleep, it shows more upregulation than downregulation in these PVT mu opioid receptor expressing neurons. So you can see many more significant transcripts were upregulated in the morphine group than downregulated. Very few were downregulated. But what does this really mean? How can we use these top hits to understand what's going on? So instead of looking at each gene individually, we did more of a pathway analysis, and we looked at all of the pathways in the KEGG database, so the Kyoto Encyclopedia for Genes and Genomes. And based on the genes that were upregulated in the morphine group, we found 15 significantly enriched KEGG pathways. And actually the fourth most significant enriched pathway was for circadian entrainment. None of the gene sets that were downregulated in the morphine group produced a significant KEGG pathway. But this was really interesting to us that circadian entrainment was so significantly enriched based on the sleep phenotype that we had seen previously. And this is an enrichment plot for that circadian entrainment pathway. So each spike here is a gene in the pathway, and you can see a lot of them are condensed here at the leading edge, where it means that each of these genes were significantly upregulated in the morphine group compared to the control group. And then just another way of looking at it, this is the entire circadian entrainment pathway. And you can see that the majority of these genes are upregulated in the morphine group, denoted in red, and a few are downregulated in the morphine group. But the majority of these genes are significantly altered from 11 days of chronic morphine in the drinking water. And this is hard to see at this point, but just a heat map of all the genes in the circadian entrainment pathway. And above this green line is the ones in the leading edge that were significantly upregulated in the morphine group. So now that we know a little bit about the profile of these neurons that popped out on the immediate early gene screen, we wondered if we could take these neurons that are obviously have a circadian mechanism, and if we could manipulate that and change the circadian behavior of mice while they're on opioids. So what we did is use chemogenetics. So we used the same Moore-Cree mouse that we used for the translating ribosome affinity purification, and you can see really dense mu-opioid receptor expression in the PVT, which is another reason that this brain region stood out to us. And we injected a virus that drives the expression of Cree-dependent inhibitory dread. So only in cells in the PVT where we inject that have Cree recombinase expressed in the mu-opioid receptor expressing neurons will we be able to inhibit these neurons with this HM4D DREADS construct. So you can see almost all of the neurons that express mu-opioid receptors also expressed our DREADS construct. And when we inject CNO, which is a normally inert ligand, but in the case of a DREADS construct, it will actually inhibit the neuron from firing. This decreased our CFOS expression in the PVT after morphine. So we repeated this increased FOS expression after a morphine injection and could block it with CNO. We also found that in an acute locomotor test for 20 minutes after the administration of morphine, our DREADS construct significantly decreased locomotion for these two 10-minute bins. So this gave us a little bit of faith that we may be able to see a behavioral phenotype from this Cree-dependent DREADS construct. So now, because our sleep phenotype was most pronounced during the dark cycle on the 11th day of morphine, we wanted to see if manipulating these PVT mu-opioid receptor expressing neurons would change their sleep on the 11th day of morphine. So we did the same sleep paradigm where we gave 11 days of 0.5 mg per mil morphine. And then 1 hour into the dark cycle on the 11th day, we injected CNO to see if that would change their sleep behavior. So the sleep recording here was done in a different system. The first exploration we did of sleep was with electroencephalography, which is really the gold standard for recording sleep and the most sensitive and can detect different stages of sleep. But here, because of the power we needed with enough animals to really scale this up to detect significance and run lots of animals and have the proper DREADS controls, we used a non-invasive sleep box that uses infrared sensors on the top of the cage that detects inactivity. And we have a program where enough inactivity has been shown to correlate well with EEG measures of sleep. So we measured sleep with what we call the COMPASS boxes, or continuous open mouse phenotyping and sleep system. So this is not invasive like the previous experiment was, but we saw the same phenotype where morphine in the solid bars increases the wake time only in the dark cycle by the 11th day of morphine here. And again, we saw no phenotype in the light cycle. And so on the next slide, I'll focus on this last day of morphine when we inject CNO 1 hour into the dark cycle, either with our DREADS construct or without. So again, same experiment here. In this case, we either have the HM4D DREADS construct or mCherry as our control, so we can give CNO to both groups of animals to account for any effects of CNO. And this top two panels just focuses on the last day of morphine. So day 11 of morphine, And then we inject CNO 1 hour into the light cycle at 8 p.m. And again, throughout the last light cycle, we see no differences between any of the groups. In the dark cycle, as expected, the solid lines of morphine groups have increased wakefulness during the dark cycle, as we had seen previously. This black bar is our M-cherry control. And we find that when we inject CNO at 8 p.m., we actually decrease the wakefulness only of the DREADS morphine group here. And this decreases the wakefulness down to the control level just for about 3 or 4 hours, which is the approximate half-life and when you see effects of CNO behaviorally. So when the CNO is no longer working, they cluster back together and show this increased wakefulness throughout the end of the dark cycle compared to the saccharin controls. Another way to look at it is to look at this entire 12-hour dark cycle period and compare it to the day before when no CNO was given. So in our mice that never received morphine, these are our saccharin mice. Our DREADS injected mice had the exact same wake properties from the second to last day to the day when they got CNO. So CNO did nothing for our saccharin controls, even though it did inhibit the neurons. But only in the morphine group do we see a significant decrease in wake time from the second to last day when they had morphine to the last day that they had morphine with CNO injected. So about 8 out of 10 mice had this significant decrease in wake time only in the morphine group, only in the DREADS group and not in the control M-cherry group. Almost all of the M-cherry mice looked exactly the same from the second to last day to the last day. So this was really interesting that we could take this genetic subset of neurons in a specific brain region, and only when they are inhibited do we see a sleep phenotype only in the drug group, not in the control group. So this is the first that we know of that has been able to change opioid-induced sleep disturbance without affecting general sleep at all. So our conclusions and future directions from this study, we showed that morphine disrupts sleep in mice, specifically at the end of the dark cycle. This is concomitant with an elevated neural activity profile in the PVT. We found that morphine upregulates the circadian entrainment pathway in these PVT mu-opioid receptor expressing cells, and that these cells are specifically required for the morphine-induced sleep disturbance. And in the future, this project could go forward by testing the directionality. We could excite these neurons instead of inhibit them. We could further investigate those specific genes that came out of top hits from our TRAP sequencing study. And we could treat sleep with insomnia drugs, and maybe that could decrease the negative effects of withdrawal and help mice or humans remain abstinent. And in fact, they're undergoing clinical trials right now with Belsomra, which is an FDA-approved insomnia drug, to see if that can extend opioid abstinence in patients. So thanks everyone for your attention, especially the funding of the T32 group. And many, many people have helped out without this project throughout the way, and especially to my mentor, Julie, who not only helped with the T32 grant, but gave me great mentoring for 6 years throughout this project. So thanks, everyone.
I had a really quick question. This is because of my naivete about these time courses, these things. You go back a few slides, the one where you were talking about things being maximal around day 11.
Yeah.
And so it's sort of parameter specific. I noticed it's going to drop down or not. Is this CNO?
Where are you looking? Right here?
But it is the CNO administration that's dropping it down in those 2 days.
Yeah, so
it could continue to, this is my question, does it continue to go up? Or what is it?
yeah, so we actually haven't looked beyond 11 days. In our EEG study, we looked only on the 11th day, so we didn't really know what happened in this middle portion. And I guess we find here it really takes until day 9 or 10 for this phenotype to emerge.
Which is really interesting in terms of just, I'm always trying to think about the analogy, obviously, in terms of when things are maximal, and it sometimes seems to take weeks and weeks of people's sleep to re-regulate after having stopped opioids.
Yeah,
After too long,
that's true. And
sometimes less.
yeah, exactly. So we just focused on chronic morphine. They don't really have much room to go up further because there are 720 minutes in a dark cycle.
Right, right.
So I don't know when it would stabilize or go up or go down, but yeah, definitely in the literature, even after you stop, your sleep is disrupted for we don't even know how long. Yeah.
Keep asking what else would you like to? I think Ray, you're up next, right? Okay. I don't know, is anybody like logged on?
I didn't yet.
Oops, okay. So. So are you zooming it? There's Ray. I'm just curious if you're logged on, if there's chat questions, you could be our chat. Okay. Yeah, can you see that on your Zoom? Interesting. All right, so let me. Yeah, this. Oh, there you are, Liz. All right, so let me share screen. There we go. And then share. All right, so that's what we have to do. And then go into PowerPoint. Okay, right. It is fourth year neuroscience student in Smith class. And she also already has a paper, molecular paper, and has done a great job on her thesis.
Thank you. All right, so today I'll be talking about my main project. Stand up. Okay. We'll set the precedent. He wants me to stand. Yeah, he deserves it. He's earned the right.
Oh, I get it.
All right, so today I'll be talking about my main project, which is looking at the role of interpeduncular nucleus glucagon-like peptide 1 receptors in fentanyl reinstatement. So our lab is really interested in exploring the glucagon-like peptide 1 receptor or GLP-1 receptor system. Glucagon-like peptide 1 is an endogenous neuropeptide and satiation hormone that's released both centrally and peripherally. And it's mostly been studied for its role in regulating both homeostatic and hedonic feeding. And actually, GLP-1 receptors are FDA approved for treating type 2 diabetes and obesity because of their role in this regulation. But what's really exciting here is because GLP-1 receptors are expressed throughout the mesolimbic dopamine system, and that's how they cause these effects on hedonic feeding, we can actually manipulate that system in order to affect substance use disorders. So there's been several studies looking at this that have found that GLP-1 receptor agonism is effective in decreasing alcohol, nicotine, and cocaine taking. However, when we look at opioids, this picture gets a little bit more complicated. So there's recent studies from our lab and others that show that GLP-1 receptors are sufficient to GLP-1 receptor agonists are sufficient to decrease oxycodone taking and seeking as well as heroin seeking. But there is one study that shows in mice, GLP-1 receptor agonism may not be sufficient to attenuate remifentanil self-administration. So this raises the question, is the ability of GLP-1 receptor agonists to decrease opioid taking dependent on the type of opioid that's being self-administered? And this is particularly important because fentanyl and its analogs are what we see driving this opioid overdose epidemic currently. So our lab looked into this question with a recently published paper where we looked at the effect of systemic GLP-1 receptor agonism on fentanyl reinstatement. So just to orient you quickly, it's active lever. No, it looks normal on this one. Yeah. Anyways, yeah, so on the y-axis, we have total lever responses during a drug plus Q induced reinstatement test. On the left side here is the active lever and the right side is the inactive lever. So active lever is these drug seeking responses. You can see as we inject larger doses of Exendin-4, there's this dose-dependent decrease in fentanyl seeking behavior. So this is really exciting. It indicates that GLP-1 receptor agonists may be a potential treatment for fentanyl use disorder. But what we still don't understand is where in the brain Exendin-4, the GLP-1 receptor agonist, is acting to cause these changes. So when I started to look into this question, I became interested in a region called the interpeduncular nucleus. So the IPN has been mostly investigated as a site where limbic signals are put together and adjust dopaminergic reward signaling. And I became interested in the IPN for a few different reasons. So firstly, the literature shows that GLP-1 receptors are expressed in the rat IPN, and I confirmed that with my preliminary data here. Secondly, there's evidence that systemic administration of a GLP-1 receptor agonist activates the rat IPN. So this is a slice of brain from a rat who received a systemic administration of Exendin-4. And these green dots here indicate FOS-positive neurons that were activated after this injection. And then finally, there is some evidence that the IPN plays a role in opioid-mediated behaviors like morphine self-administration, morphine withdrawal, and dopamine sensitization to repeated morphine. But the role of the IPN in these behaviors is not completely clear. So based on all of these findings in the literature, I hypothesized that intra-IPN Exendin-4 would be sufficient to attenuate fentanyl seeking in male and female rats. So in order to explore this question, I had rats go through 21 days of fentanyl self-administration, then through extinction until they ceased responding on the active lever. And then they had this drug plus Q induced reinstatement testing. So here's the results from the male rats. Again, we have the total lever responses, active and inactive lever. And then here we have the total infusions, which is just infusions of saline that they received during the reinstatement test. Again, you can see that there's this dose-dependent decrease in fentanyl seeking as we inject higher doses of Exendin-4 into the IPN. And when we look at females, we also see the same thing where we have this decrease in fentanyl seeking with IPN Exendin-4. So one thing that's important to consider when you're thinking about using GLP-1 receptor agonists as a treatment for substance use disorders is that in the clinic, these have been shown to sometimes induce nausea and malaise. So I wanted to investigate if maybe these were affecting food intake, causing nausea in these animals. So during each of these reinstatement tests, I measured, here's 24-hour food intake, then 24-hour change in body weight, 24-hour water intake, and 24-hour kaolin intake. So kaolin, non-nutritive substance that animals eat as part of a pica behavior, so we can use this as a proxy for nausea. And then in both males and females, we did not see an effect of IPN Exendin-4 on any of these measurements, which is promising. It indicates that probably they're not feeling too nauseous, that it's affecting their eating or anything like that. So then I wanted to understand more about where in the IPN and what circuits are actually underlying this effect. First, I phenotyped GLP-1 receptor expressing neurons with RNA-scope and found that over 90% of the IPN GLP-1 receptor expressing neurons are GABAergic. And I also wanted to look at where in the IPN GLP-1 receptors are expressed because we have there's specific subregions of the IPN that have specific projection sites. So first I looked at it along the anterior to posterior coordinate. And you can see here, there is definitely a greater expression of GLP-1 receptors in the more posterior IPN, which is also supported by the literature. However, when I then switched to look more at the subregion specifically, I didn't see a significant difference between these regions. But I noticed just while looking at the slides in this quasi-subregion called the IPN dorsolateral, there was definitely a much higher expression of GLP-1 receptors. That was probably where the majority of GLP-1 receptors were in the IPN. And I thought this was particularly interesting given that the IPN-DL has been shown to send GABAergic projections to the laterodorsal tegmental nucleus, which also has been indicated to have some role in opioid seeking. So in order to investigate if GLP-1 receptors are actually expressed on this GABAergic projection to the LDTG, I injected fluorogold into the LDTG and again did RNA scope and found that GLP-1 receptors are expressed on this GABAergic projection to the LDTG. And I also found mu opioid receptor expression on these same projections. So altogether, this indicates this might be a circuit underlying the effect of exendin 4 and in the IPN on conventional seeking. In conclusion, I found that IPN exendin 4 is sufficient to attenuate fentanyl seeking behavior in male and female rats without affecting body weight, food or water intake, or Kalin. And I also found a potential circuit underlying this effect where GLP-1 receptors and mu opioid receptors are expressed on GABAergic IPN neurons that project to the LDTG. So this is my hypothesized circuit, given the information we have at the moment. So I'm predicting that possibly with the administration of fentanyl, this binds to mu opioid receptors on GABAergic IPN neurons, which then decreases the GABAergic input onto excitatory LDTG projections to the VTA, thus allowing opioid-mediated dopamine release. And when we administer exendin 4, I'm hypothesizing that exendin 4 binds to the IPN to excitatory GLP-1 receptors to activate this projection and thus inhibit opioid-mediated dopamine release. So moving forward, I have a few experiments I'm working on to look into these questions. Firstly, given that the IPN has been shown to have an important role in anxiety and aversion, I want to look at the effect of GLP-1 receptor activation on these measures. Secondly, I'm going to use fiber photometry to measure calcium activity in IPN GABA neurons during fentanyl reinstatement and exendin 4 treatment. And then finally, I'm going to use chemogenetics to activate and inhibit this circuit during fentanyl reinstatement and exendin 4 treatment to further understand its role in this behavior. So yeah, just thank you. Rob. Greg. Thank you.
And an education question for me, on the Kalin taking, is it different for males and females
Like in
in
general?
terms of these
Are these-
nausea related analog? It looked like the fuchsia bars were taller than the green bars on the Kalin.
Definitely might have.
I'm sorry, but there.
Yeah, yeah,
So
it does look like the females were taking more.
I just wonder if there's a bigger, and I don't know the medications, are there bigger nausea responses to these in humans and women than men? That is no.
I was wondering what didn't waste this space.
Oh. It's a mystery. Thank you, Riley.
I guess I was just wondering real quick, would it be so bad for these drugs to cause some sort of nausea malaise? Wouldn't that be a good, thinking about antabuse, wouldn't that be a good sort of conditioned association to like feel sick and I don't want to take opioids? Sure. Are there any chat questions? If I'm the Greg you're mentioning, oh. Okay.
You found the grid.
Okay, sorry, Greg. Okay, so next share screen.
Good comment.
My comment is, I am no longer referring to it as a comfort and I'm calling it an alternative.
Interesting.
The more you read, you're.
That's a good point. Are you, have you seen that from,
Well, you read the history of the staff protocol.
oh, yeah,
They've been brought in.
yeah,
It's not really a question of whether. Now that we all know what it can do. Things that come out of that authority.
right. Yeah, that's,
I did have another question more too, is this, is just, what's your thought about remnant versus techno into basically, so it wasn't always working with remnant?
Yeah, so I would say there's a few issues with the design of that study, so I wouldn't necessarily say that it's not effective in fentanyl. I think there are functional analogs, they like self-administration, occurs. Yeah.
Just curious, because of course, you know, next week's fentanyl, everybody's making analogs as quickly as they can, they're cheap. Don't have to look for a plant, no crop, or trade wind or anything. So it's really a mess, and I just was curious about that and consumer out of that. Thank you.
our next speaker is Julia Ferrante, who is a rising third-year pharmacology graduate student, and I was fortunate to have her join my lab. And she's going to be telling you about our neonatal opiate withdrawal model.
Yeah, thank you. So today I'll be sharing some of our work just with our model in general, and then specifically my project, which is attempting to characterize the role of activated microglia in neonatal opioid withdrawal syndrome. So as I'm sure, as we all know in this room, there is this ongoing opioid genocide, and there are really a lot of stark consequences of this, but sort of one of the more understudied populations are these babies who are born to opioid users. And so as the number of opioid users have grown, this other population that still, remains to be studied is also growing at this really staggering rate. So when babies are born to mothers who use opioids throughout their pregnancy, they're at risk of developing neonatal opioid withdrawal syndrome. And this is a well-characterized just number of somatic withdrawal symptoms, including things like high-pitched crying, irritability, seizures, cardiac issues, GI issues. And these usually arise about 24 to 48 hours after birth. And there's no real treatment for NOWs. Some hospitals do choose to treat with opioid tapering or buprenorphine, but I'm sure, as you can imagine, this is pretty controversial and also tends to be very hospital specific. So there really is no standard treatment. And although these acute somatic withdrawal symptoms are really well characterized, the more long-term consequences of NOWs are still relatively unknown. However, there have been many attempts to sort of characterize these clinical outcomes, although these are really limited. First, by a lot of confounding external factors, including things like maternal polysubstance abuse, socioeconomic status, poor maternal care. And there's also a lot of difficulty in conducting these longitudinal studies. And so for this data I'm about to present, this is really limited up until the adolescence time point. But they have been able to show things like low birth weights, impaired physiological development, as well as changes neuronally from reduced white matter and things like the basal ganglia, thalamus, and cerebellum. There are also reports of attention and behavioral deficits, as well as cognitive and motor deficits. And, a study that was really interesting to me and more related to my project, there was one study that came out that showed an increased rate of infections and parasitic diseases, as well as an increased rate of hospitalizations from infections in the first 3 years of life in babies who are diagnosed with NOWs. So this indicates that there's some potential immune modulation happening in babies who are diagnosed with this disease. And since there is so many difficulties in conducting these clinical studies, there have been a lot of attempts to model NOWs using rodent models. But as you can imagine, there's a lot of models and there's a lot of variables to consider when developing one of these models. And so one of the first things to consider is the type of opioid that each model is using. So are they using morphine, which is the prototypical mu agonist, or things that are seen more clinically, so fentanyl, heroin, oxycodone. Additionally, the duration of opioid exposure to both the dam and the pups, as well as the route of administration of the opioid. But these models have been able to recapitulate some of the changes that we've seen clinically, so things like physical and neuronal development, cognition, behavior, and additionally in opioid behavior. But these experimental outcomes tend to vary widely between models, and even two models can show completely contradictory results just depending on some of these variables I previously mentioned. But a few studies have also gone on to investigate the effects of NOWs on immune functioning and specifically neuroimmune functioning. And this is especially important because opioids are known modulators of the immune system. So when a person takes opioids, they affect the immune system in two very distinct ways. So in the periphery, opioids tend to be immunosuppressive, and they affect things like cytokine production and NK cell activity, antibody production, and lymphocyte proliferation. But within the central, within the central nervous system, they have pro-inflammatory effects, primarily acting via activating the microglia and then causing the subsequent release of pro-inflammatory cytokines. And this is pretty well established in just adult models of opioid taking, but this has not yet been characterized in models of early life opioid taking and subsequent withdrawal. So this leads to my experimental question: how does early life opiate exposure and withdrawal modulate neuroimmune functioning? And my hypothesis is that early life opiate exposure and withdrawal increases activated microglia, resulting in mouse symptomology and long-term neuroimmune and behavioral consequences. So in order to answer this question, the Blendy Lab has developed our own model of NOWs, which we coin as the 3-trimester model. And we call it this because we have sought to recapitulate the entire developmental equivalent of the human pregnancy, including all 3 trimesters. This is important because, as you can see up there, the gestational period of mice is a lot shorter than the gestational period of humans. So if we were to just deliver opioids during this gestation period, we'd only be getting the equivalent of the first 2 trimesters of human pregnancy. So in order to get this equivalent of the 3rd trimester, we have to go further and look at these first 2 postnatal weeks in mice. And this is a really critical neurodevelopmental period in mice because this is where things like synaptogenesis as well as microglia maturation occur. So with our model, we deliver opiate exposure throughout all of gestation and then those first 2 postnatal weeks. So with our model, first dams are implanted with subcutaneous mini pumps that release morphine at a dose of 10 mg per kg per day when they're allowed to mate. And then while the pups are in utero, they are exposed to morphine in gestation. Then once they are born, we cross-foster these pups to opioid naive mothers, and this reduces any effects, confounding potentially of poor maternal care. And then they receive twice daily injection of morphine at that same dose of 10 mg per kg. And at postnatal day 14, they receive their final morphine injection and they're allowed to go through spontaneous withdrawal. So just to validate our model a bit, first we looked at developmental milestones and sought to see if we could recapitulate some of these developmental delays. So in 4 tasks of forelimb grasp, ambulation, surface writing, and eye opening, we found that the mice who were exposed to morphine had increased latencies to reach all these milestones. Additionally, we also looked at weight gain over development, and we found that the mice who got morphine, both male and female, had decreased body weight up until PND 15. And importantly, since we are studying neonatal opioid withdrawal syndrome, it's very important that our mice are going through this robust withdrawal. So first, we looked at withdrawal at 3 different time points, and our first test was thermal hyperalgesia, which we used for the hot plate test. We found at 8 hours and 24 hours, there's a decreased latency to lift paw. We also looked at ultrasonic vocalizations, which were increased at all 3 time points, as well as somatic signs of withdrawal, which include things like wall jumps and climbs in the mice, and these were increased at 24 hours. So going forward, we only measure withdrawal at this 24-hour time point. So for the first portion of my question, we want to just look at this early life acute period following the 3-trimester model and seeing if there were any changes occurring in neuroimmune response. So this sort of came out of this very initial pilot data from the Blendy Lab, which didn't actually use the 3-trimester model, but it did use this modified exposure period, which is just morphine exposure from PND 1 through 14, followed by precipitated naloxone-induced withdrawal, just to see if we get this initial robust immune response. And indeed, when looking at IHC for IBA1, and IBA1 is a marker of microglia, we found that the mice who were exposed to morphine had a 30% increase in the number of microglia. We also did qPCR for some immune-related genes and found that C1q, which is part of the complement pathway, as well as CD68, which is a macrophage marker, were both significantly increased in expression. And this is all in the cortex. So it was really important that although we saw this in this sort of more, I guess, activated model where we had this precipitated withdrawal and then this modified exposure paradigm, we saw these neuroimmune changes. But if going forward with our model, we could actually recapitulate these with the full 3-trimester exposure. So we sent some brains over to our collaborators at Mount Sinai. And when looking in the ventral striatum, we found that there were increased microglia in the mice who were exposed to the 3-trimester paradigm, again, at that PND 15 time point. But this led us to another question. So we have these mice who went through this model, but our further question here was, are these neuroimmune changes being induced by just this early life opiate exposure on its own, or this combination of the early life opiate exposure and the withdrawal? So to answer this, we put our mice again through the 3-trimester exposure model, but then separated them into 2 groups. So in our first group up at top, we gave them their final morphine injection at PND 14, and then 2 hours later, they were sacked and we collected their brains. So presumably morphine is still in the system and they're not yet undergoing withdrawal. In our second group, just like in the standard 3-trimester model, we gave them the last morphine injection on PND 14, and then 24 hours later, they went through withdrawal testing and we quantified that. And then we collected their brains at PND 15, 24 hours later. So these mice did go through withdrawal. And all this still kind of preliminary data, I'm still analyzing it, looking at some different brain regions, but just starting the ventral striatum, we measured IHC again for IBA1 using our marker of microglia. And in the bottom two panels down here, these are morphine exposed mice. So on the left here is the mice who did not go through withdrawal, who were stacked at PND 14. And on the right is the mice who were stacked at PND 15, who did go through withdrawal. And from our quantification, we found that only the mice who had the opioid exposure and withdrawal had significant increases in microglia. So some conclusions for my portion, which is just looking at the early life period. First, we were able to develop a validated rodent model of NOWS. Second, we were able to find that early life opioid exposure and withdrawal results in increased microglia in the ventral striatum, but just early life opioid exposure without withdrawal may not be sufficient to produce any of these neuroimmune changes. So some future directions for this portion. First, I want to go further and examine microglial morphology, since just number on its own is only telling us sort of half the story of what's actually occurring. Going on to analyze other brain regions, since so far we just have data on cortex and striatum. And then just sort of going beyond microglia on its own, looking at some changes in cytokines as well as immune-related genes at the same time point. And further, just beyond the central nervous system, looking at some systemic immune changes as well. And so far, all of our data is just at baseline in this immune response. So looking at how these mice may also respond to an immune challenge. So for my second portion, now that we've established these baseline sort of early life neuroimmune changes, this led us to our next question, which is looking at if these immune changes can persist up until adulthood. So in order to answer this, I again put the mice through a 3-trimester model, but they are allowed to grow up into adulthood with no further drug exposure. Once they're in adulthood, they received injections of lipopolysaccharides, also known as LPS. And this is a part of the bacterial cell wall. And so when injected into animals, it produces this really robust and well-characterized immune response. And along with this, 24 hours later, I collected their brain to see if we could see any changes. Again, just so far looking at microglia and seeing those changes in that level. And I also looked at somatic signs of sickness, as well as got readouts of activity data using the compass boxes that Daryl very nicely explained for us earlier. And so just to look at baseline adult immune response to see if these changes could persist, I was able to measure this by using my control animals. So these are animals who just got saline injections in adulthood. And when comparing the control and 3-trimester mice, we found that there were no changes in expression of IBA1. But when the mice received an injection of LPS, it was only in the mice who had the 3-trimester exposure had an increase in IBA1 expression even over the control mice who also received the same LPS injection. So this indicates that there may be some early life immune changes. These do not persist up into adulthood, but given a response of some immune challenge, there may be this potential hyperactivation of the immune system. Additionally, while I had these mice, I also probed them for somatic signs of sickness, but I found that there were no changes. So one of the first things was body weight, and all the mice did lose a similar amount of body weight. They had a significant decrease regardless of their prenatal treatment. Also looking at body temperature changes, all the mice who received LPS had a hypothermic response, whether or not really they received saline or morphine. Although I guess almost interestingly, if you do look at the axes here, the females did have a much lower response than the males did. So potentially undercutting some sex differences, although this didn't seem to be changed whether or not they had morphine or saline in utero. And also looking at COAT score, there were no changes. So one of the other things I looked at was activity data. But again, this is not significantly altered in the 3-trimester mice, and all mice who received LPS did have this decrease in activity data over time as a result. Although it is important to note that this is my first experiment with it, with using the LPS, and I was sort of experimenting at the time. But when I injected it at this time point, I did only inject during the light cycle. So there is potentially this confounding factor of like this floor or ceiling effect where the mice should have been asleep anyway. So especially at these early time points, which are maybe more critical for analyzing the sickness behavior, I wasn't really accurately measuring that there. So in further studies, which I'm still quantifying right now, I injected into the dark cycle. And so potentially maybe there'll be some changes there. So some conclusions. Found that opioid-induced early life increases in microglia may normalize in adulthood, and the 3-trimester model can cause increases in microglia expression only after a bacterial immune challenge. But somatic signs of sickness are not exacerbated by previous exposure to the 3-trimester model. So some future directions. First, I'm looking at different doses of LPS. So far, what I've done is use 2 mg/kg, which is a really high dose. You can start a response at as little as like 0.1 mg/kg. So could potentially, again, be getting some sort of ceiling effects there. Additionally, the data so far in this adult time point is only done with qPCR for IBA1. So I want to go further and do IHC. And then again, you can also get a better picture of the microglia morphology there. And going just further beyond the microglia, looking at this analysis of cytokines and immune-related genes. as well as further expanding this just to a whole broad systemic immune response, since we do know that opioids also have this immunosuppressive effect in the periphery. Also looking at some long-term behavioral effects other than just sickness response. And going beyond that, so since I have established at least some evidence for these changes in microglia, but I did not see these changes in sickness behavior, it's possible that I'm just sort of cornering myself into a hole with just correlating immune response and microglia, but microglia have a lot of other roles, right? So one of the other things we can look at is some of these other potential roles of microglia being changed, including things like synaptic plasticity. So some other routes also to investigate. Thank you to everyone in the Blendy lab. Also Emma, who helps, she's online right now, who helps with some of these experiments during her rotation. Sophia, who was an SUIP student we had, and who really helped me out during this summer. Of course, to the T32, and for everyone for being here today, listening. Hi, Heath. Yeah, so it's interesting because our behavioral data is interesting. So one of the first things we looked at was just some of the baseline behavior. So things like affective behavior. I think I have it somewhere here, like opioid seeking behavior. And with all of that, so yeah, none of the affective behaviors were changed. This is all in adulthood. And none of the opioid reward behaviors were changed. So this does indicate that that's not changed. And especially since the striatum is this like big hub for, drug reinforcement, it is interesting that we don't see these concurrent behavioral changes, but some of the other things that we have seen potentially behavioral changes in our social behavior. And although we haven't perfectly validated the assay yet, that is sort of another route we're looking at, but it doesn't necessarily seem to correlate with the behaviors that we think it would, so. It could, yeah, I mean, it's really limited so far just because I only have data from striatum and then some from cortex too. So whether this is a full brain wide phenomenon or it's just from these regions that we've just been kind of picking and choosing, I think it would take a little further and be a little more interesting to see if we do see changes here, but not here, and then maybe pinpoint from that what's actually going on.
They're not more sick.
Yeah. Yeah, they didn't appear to be more sick.
So, and then there's also the virtual, so there's a like we send one of somehow was talking not.
Potentially. Yeah. Yeah, I mean, I was really hoping they would get sicker. I think that would have made for a nice, better picture, right? It would have made a nice, easier hypothesis, but if that does hold true and there still are those changes, but there's no, correlating behavior, then I think that it's worth looking for other routes than just immune response changes.
This is actually a healthy response. So yes, so it's like the stable in every single adult. If that didn't happen, then they.
Yeah, yeah. It's interesting that it does seem almost like to be primed to react in this way, but I just haven't gotten that next piece yet of what it's doing. Yeah.
I know, I was just going to tell you. They are so slow. Yeah, that was slow. Very smooth. And we're well ahead of time, which is good. Well, that was because she was so. Cool.
All right.
Yeah, so our next. Oh, I don't know.
Yeah, no, it's, yeah, this microglia stuff could be very interesting. And we also haven't looked at recovery, right? And do we have the right sickness, quote unquote, sickness behaviors? Where are you, Adrian? I know you're here. Here we go. Whoops. Okay. Share screen. And oops. Sorry, the There we go. Next, our final pre-doctoral speaker is Adrian Joe. And Adrian is also a pharmacology graduate student rising 4th year, who has joined Greg's lab and is co-mentored by Greg and I. And she's going to be talking about a new project that actually we did that a year ago, about circuit mapping.
Cool. Thanks, Julie. Hi, everyone. I'm Adrian. And so today I'll be presenting my project on mapping mu opioid receptor expressing circuits across structural topologies. And as Julie mentioned, I actually recently just joined Dr. Julie Blendy and Dr. Greg Corder's labs. So this presentation will be an overview of the project that I'm now working on. So as we're all aware, opioid use disorders have utterly devastated the US, with around 3 million suffering from an OUD as of 2022. Additionally, opioids account for roughly 75% of all drug-related overdose deaths. And so this over here is a bar graph that was published from the CDC that depicts the national overdose deaths involving any opioid, whether that's prescription opioids, heroin, or synthetic opioids like fentanyl, among all ages from 1999 to 2020. So just to point out some key numbers, we could see that in 2010, the number of deaths attributed to overdoses from opioids were around 21,000. And then only in 7 years, it more than doubled to over 47,000. And then in 2020, it was over 68,000. And unfortunately, COVID has only further exacerbated the number of overdose deaths that are attributed to not only drugs in general, but specifically opioids. And so overall, these tragedies just highlight this need to have a deeper understanding into the opioid neurobiology that has a pivotal role in influencing opioid use disorder states, in order to kind of use this research to think about new therapeutics to treat opioid use disorders. And so with that said, we know that opioids primarily produce their analgesic, rewarding, withdrawal, and dependence-like phenotypes by acting and stimulating upon the mu opioid receptor, or MORS. And so mu opioid receptors were actually discovered roughly 5 decades ago in 1973. And although we've had this understanding about MORS for roughly 5 decades, what's interesting is that the actual mu opioid receptor circuitry throughout the various stages of the opioid use disorder cycle actually remains unknown. How do these actual opioid circuitries adapt and shift over time? And so with that said, kind of the fundamental question we have here is, how do mu opioid receptor expressing neural circuits facilitate and maintain dependence and withdrawal? And so we know that there are different neuroadaptations that are associated with these physical and cellular dependence mechanisms, where you have a neural network that's able to only function homeostatically in the presence of this opioid. And so here I'll just show a mechanism of action that's kind of like the classic MOA of opioids that involves a disinhibition of GABAergic neurons in the VTA. And so normally we have this cortical control of firing of these GABAergic neurons in the VTA that inhibit these dopaminergic neurons that then project onto the NAC, or the nucleus accumbens. However, in the presence of an opioid, as this opioid binds onto its mu opioid receptor, this action actually inhibits GABAergic neurons, which leads to the GABAergic neuron being unable to subsequently inhibit these dopaminergic neurons that project and release onto the nucleus accumbens. So, what is the overall net effect of this? It is that there is an increase in dopaminergic firing and an increase in dopaminergic release. And when we think about kind of the long-term effects of this mu opioid receptor mediated inhibition, there are a series of different neuroplasticity related phenotypes that could result. And so one of them being, this chronic stimulation of more mediated inhibitory cascades can lead to different transcriptional programs where, in the state of withdrawal, when the drug is no longer there, this can lead to an increase in excitatory drive and neural hyperactivity. And over time, this could induce these alterations in extensive circuits that influence these network range dependence mechanisms. However, this is kind of like only one aspect of all the different sorts of neuroplasticity related mechanisms. And it gets even more complex when you think about the permutations that result from all these different time points of development with OUD, with respect to all the different brain regions, with respect to all the different types of cells that may, all kind of synergize to involve different manifestations of opioid use disorders. And so with that said, we know that there are kind of like central hubs that have high expression of mu opioid receptors. And so this is just kind of like a classic, well-characterized reward circuitry of the VTA projecting onto nucleus accumbens, and kind of other regions that we know that have high mu opioid receptor expression include like the VTA, the nucleus accumbens, the thalamus, the cingulate cortex, etc. And so the fundamental question we have here is, for all these regions that have mu opioid receptors, where exactly do they even project to? And how do these projections of more expressing neural populations evolve over time, over the course of opioid exposure? Therefore, kind of the primary aim of this project is to precisely map out the shifting connectivity of opioidergic networks throughout various stages of the OUD cycle with respect to the different regions. And many studies have focused on a region or two regions. However, for this aim, we really want to take into account all the different dynamic processes that come together that enable a state to go from one drug-induced state to another. And so with that said, we will be focusing on numerous kind of core regions involved in the OUD mechanism. And so with that said, we will be using this paradigm. For morphine exposure, we will not be using intravenous self-administration. Instead, we'll be using these 3D printed licometer devices. And so this will enable the mice to have 24-hour access to morphine from the comfort of their own home. And so some challenges that we could circumvent through using this is the challenges from IBSA, which involve after surgerizing the mice and inserting this catheter, it could influence processes like tissue injury-induced inflammation or pain processes that can have an effect on, the endogenous opioid system. And so with this said, we're just using this 3D printed model where they can just drink morphine at their own disposal. We'll also be interested in opioid use disorder with respect to different time points. So opioid naive, acute exposure, chronic exposure, and then abstinence. And like I mentioned, we have this emphasis on looking at opioid connectivity from different core regions. And so the nucleus accumbens, the central amygdala, the ventral tegmental area, and it's boxed here in red because this is actually our first kind of region of interest that we'll be exploring. And then the ventrolateral periaqueductal gray. And so for this project, we want to specifically interrogate cells that have the mu opioid receptor. And so how exactly do we do this? We can actually use mice that are specific to the mu opioid receptor. And these are OPRM1 Cree mice that have been well developed and characterized in our lab, in the Blendy lab. And so the mu opioid receptor is encoded by this gene OPRM1. And so for these mice, we have this Cree recombinase, or just Cree enzyme that's active only in cells expressing the OPRM1 promoter. And so what that means is that that Cree is only expressed in cells that have this mu opioid receptor. And so this is important because later on, we'll be using these viral vectors that are specific to Cree in order to specifically target these mu opioid receptor expressive cells. And so this image is just confirmation of Cree-mediated recombination. And this was done by inserting this GFP reporter into Cree, as depicted here in green. And this is in a region that we're interested in, which is the VTA. And then another image here just confirms Cree-mediated combination, but specifically in mu opioid receptor cells. And this was done through co-localization of GFP, green fluorescent proteins, to kind of code for this Cree, as well as mu opioid receptors, as indicated in this magenta. And then for this image, this is in an area with high mu opioid receptor expression, the habenula. And so then moving forward, we're interested in using these viral vectors to, number one, map out structural connections of cells that contain mu opioid receptors. And second, think about the structural plasticity of these connections over time with respect to different phases of the OUD cycle. And so we want to do this by mapping on axonal inputs. So what inputs into the VTA and outputs? What are the axons that are outputted, I guess, from the ventral tegmental area? And so one way that we could look at the axonal inputs is by using this method. It's called rabies virus or RAB-V input mapping. And this will be done again in our OPRM Cree mice. This is to specifically target our mu opioid receptor neurons. And so this method will enable the identification of neurons that are projecting onto a genetically defined starter population for us, the ventral tegmental area. And so first, we'll be injecting these helper viruses, these AAVs, into our core region within the VTA. And so these AAVs will be specifically targeting cells that have the mu opioid receptor. And then afterwards, 5 days before we collect the brain, again, we're interested in kind of what is the neuron that's inputting into the VTA. So 5 days before collecting the brain, we'll be using this RAB-V delta G GFP virus. And why I said delta G is important is because it will have the glycoprotein G removed, which will enable monosynaptic tracing, so that when we inject it into the VTA, it will cross one synaptic step just to infect the presynaptic partner of VTA neurons that express TBA. And so we have this like cell type specificity of just looking at the more expressing cells in the VTA and kind of what is synapsing onto them. And then to address our interests in, okay, you know, what are the axons protruding out of the ventral tegmental area? We'll be using this technology called TrailMap. Kind of a mouthful, but it's tissue registration and automated identification of light sheet microscope acquired projectomes. So the goal here is to visualize axonal projections in 3D from intact, cleared mouse brains using light sheet microscopy. And so first, we'll be using these anterior grade tracers, meaning they will be taken up from the cell body and travel toward the axons to think about, okay, you know, what are the axons leaving the VTA? And we will then clear the tissue. And this is important because we want to be able to visualize these deep, complex structures, these axonal projections. And then we'll image the brains through light sheet microscopy. And then from these images, we'll be able to create these 3D volumetric representations of these identified projections. And then after that, we can actually color code each axonal projection and then use this to create heat maps of altering axonal innervation and structural connections that will show a spectrum of different synaptic strains. And then for our actual opioid dosing regimes, again, we are using these OPRM1 Cree mice. So at 8 weeks of age, we will be inserting, or rather injecting into the VTA our AAV helper viruses. And then a week later, we'll enable the mice to habituate to this 3D device glycometer in their home cage. And then a week later, so 2 weeks following surgery, we'll actually begin the different opioid dosing protocols. And so like I mentioned in the beginning, we're interested in 4 different time points. So for an opioid naive, these mice will undergo 5 days of just saccharin. For acute morphine, they will undergo 5 days of morphine at the concentration of 0.3 mg per milliliter. And then for chronic morphine, we want to use escalating doses of morphine for 21 days. So starting from 0.3 and then 0.5 and then 0.75 mg per mil. And then for protracted withdrawal, we will also be using these escalating morphine doses for 21 days. And then this will then be followed by 14 days of just abstinence. And so, like I mentioned, 5 days before we actually collect the brains, we will be injecting this RAB-V delta G, RAB-V delta G virus. And so this will actually enable us to see, you know, what are the axonal inputs with respect to all 4 of these different time points. And so in terms of just general expectations from this project, from this part of the project rather, we expect to identify differences in input patterns of mu opioid receptor expressing populations across time. signifying that there might be changes in structural connectivity throughout the duration of opioid administration and withdrawal. And from this, if we identify specific nodes that represent a disproportionately large factor in affecting the circuit, it could be like, oh, this might be a central hub that has an influence in this specific opioid state or during withdrawal. And so, yeah, just in terms of my acknowledgement slide, of course, my mentors, Dr. Blendy and Corder, everyone in the Blendy lab and Corder lab who has made my transition the most streamlined process. Very grateful to be in this position, as well as all of our collaborators on this project. So we have Drs. Bassett at Penn, Eustace Kepschel at Johns Hopkins, and then Kevin Beyer at UC Irvine. Of course, thank you to NIDA and the NIH for funding this research, as well as the pharmacology graduate group led by Julie, also with Sarah Squire. So yeah, with that, thank you so much.
Yeah, go ahead.
Yes, so for this, we will be using these 3D sippers, where we could actually extract data such as the number of times a mouse actually approached the sipper to lick the clinical tube to get morphine, as well as the time duration. And so there might be differences across these different phases, but we will do forced drinking, meaning that the only way they could drink any liquid is through this morphine solution through the sipper. And so I guess, yeah, that's something I definitely want to ask from previous people who've run this is, was there a lot of variability? From looking at some of the prior graphs, it doesn't seem like there was striking variability, but I think that is definitely a potential consideration. So we will be doing forced drinking. So it'll just be either one bottle of just morphine or maybe 2 bottles of this morphine solution in saccharin. Yes. Yes, so I think kind of in preparation for this, that really emphasizes the importance for these to be very or as accurate of hits as possible, and to actually ensure that we're hitting our target regions of the VTA. So I guess there's not really a way to know kind of the accuracy of my injection until the very end. And so hopefully with a lot of practice to accurately hit our region of interest, and B, running enough animals to consider, okay, if this is a mouse where the target hit was completely off for XYZ, we could exclude that and still have a substantial number of N's. Cool. Okay, thank you.
Okay, now we're moving on to here we go. Yes. This is a little complicated. Share screen. No. Then. Okay. Oh.
So our next presenter is Costa Pappas, who is postdoc in Bob Mack's lab. And one of the fun things for him this past year is that he was selected as one of 8 Penn trainees for the Advanced Therapies Intensive, a week-long program in partnership with King's Health Partner in the UK, sponsored by Penn's Institute for Translational translational medicine and therapeutics. So it was a week-long thing that you did relatively recently,
Yeah, just earlier in September.
life-changing.
It was different, yeah, as a chemist learning all this biology stuff, tough. I don't know how you. But it was very eye-opening to new therapeutics, certainly.
Well, you'll have your chance now for everybody to say, how does he do all that chemistry stuff?
Yeah.
Now for something completely.
So unusual here, all focused on opioids.
Yeah, it's not so unusual.
I guess that's so unusual.
Right. Did everyone hear me? Everybody good? Doing okay? Awesome. So thank you very much for the introduction. I will be discussing one of my main projects today. So there are two areas of interest that I spend most of my time. The first is no stranger to anybody who's been attending the P30 talks once a month. So that's the development of mu opioid receptor antagonists. And there are two main reasons why I'm doing that. One is for therapeutic interests. So that's geared towards solving this problem known as re-narcotization. And I'll talk a little bit about what that is, why we should care about it in the coming slides. The original interest from me, why I wanted to join Bob's group and do this, was actually focused more on the diagnostic purposes. So I've used my skills as a chemist to conduct structure activity relationship campaigns, make lots of molecules. And my original goal was to try and translate something into a novel mu antagonist PET tracer. And we'll get there. We'll talk about how that's going. I have a completely separate project, lots of different chemistry, much more lengthy synthesis on the development of delta opioid receptor agonists. With the key goal behind that is finding something to displace methylnaltrindole, which is currently the gold standard, but it can be a little lipophilic. It doesn't really produce reproducible time activity curves. So I'd argue there is an urgent need to address both of these thrusts in my projects. As I mentioned, we'll probably focus on the mu opioid receptor antagonist project today. And I'll start off with discussing the therapeutic applications. So this graph probably looks very familiar because it was just presented. I swear I didn't copy your presentation, which means we both shop at the same place, I guess. But to also borrow a term or phrase, yes, we do indeed find ourselves in opioid. I won't call it a pandemic or epidemic, but it could really properly be defined as a genocide if you've read any of the history of the Sackler family. And there's certainly some good books that I mentioned in my journal club that I'd be happy to recommend to anybody. But 70 to 90,000 loss of life, that is a significant number of people that we've lost. And that's just this most recent year. Now, historically, we think of this morbidity and mortality as being driven by classic opioids. So here I have just the structure of morphine. I'm a chemist, I'm a structures guy. So this is the structure of morphine. It is a useful medicine, especially providing analgesia. But through time, chemists, both proper and clandestine chemists, have found ways to make molecules that have a higher potential for abuse. And so this is morphine on the bottom. This is diacetyl morphine, also known as heroin. So structurally similar, but we can see that about 2011, this heroin really started to supplant other opioids and kind of get a bigger share of the morbidity and mortality. But recently, as we know, we've kind of been dominated. The lion's share of this carnage has been claimed by things like fentanyl. One of the key reasons is that it's actually pretty easy to make and source the materials to make these kind of illicit drugs. The burden of this pandemic or genocide is catastrophic. So 44 people die every day, and that's just from things like prescription opioids. You can imagine the number is actually much higher for things such as illicit opioids, ones with relatively no therapeutic benefit. And if you're more of a economist or capitalist, you can also think about this costing a lot of money in healthcare costs. So annually about 35 billion and 92 billion in terms of loss of productivity. One of the interesting themes or phenomena that we find ourselves witnessing in the synthetic opioid era is this concept known as re-narcotization. And it's been well documented in the literature. So re-narcotization occurs when you might encounter someone who's experiencing opioid overdose. You or a first responder could administer Narcan, life-saving medicine. They can appear fine and then subsequently slip back into respiratory depression. This was accounted in this journal. So this details a particularly deadly night in New Haven, Connecticut, where we have about 12 admits to the ICU, many of which were first attended to by first responders. And the 4 I've highlighted received an initial 2 milligram dose of Narcan to combat their opioid overdose, appeared fine, and then subsequently slipped back into respiratory depression. So pretty scary. One patient, the one I've highlighted in red, needed a continuous 4 milligram per kilogram per hour drip of naloxone to kind of sustain his or her coming out of this respiratory failure. So I would use this to argue that there is an unmet need, an urgent unmet need for the development of new novel antidotes to solve this problem that somewhat belong to this synthetic opioid era. One of the issues with drug development is that it can be both a timely and costly endeavor. So traditionally, we learned in our med chem class, it could take 10 to 15 years to get something from bench to bedside.
That's one.
Make that 20, yeah, 20 billions. Right, it's certainly not cheap. And so I've really thought recently, like, what are some ways or what are some methods we can use to kind of expedite this process? And one method is drug repurposing, which is a broad term, but the idea is we want to be able to identify a molecule quickly. Maybe it's underwent some clinical investigation, ideally preclinical, phase 1, phase 2, and for whatever reason, maybe a pharmaceutical company doesn't care about it anymore. Then I could take it and do all sorts of neat things, and thus kind of cut down on the time it would be required to get this from soup to nuts, from idea to being able to market this to, not market, but deliver this to patients in need. So there's a disconnect. There we go. All right. So if we wanted to develop an antidote to help us address this issue, some key parameters you might want to keep in mind is we'll probably want a molecule that binds potently and selectively to the mu-opioid receptor. We'd like its intrinsic activity to be non-activating, so that necessitates a non-agonist or perhaps an inverse agonist. From a synthetic accessibility, that's something I would like. I'd like it to be easy and modular, able to scale up so that we can make it cheap, provide it to people who need it. Ideally, this should get to the brain. It's been shown in lots of PET studies that I'll get to later that displacing the opioids from the brain is an important part of being able to reinstate respiratory function. And as I alluded to before, if this molecule or these molecules have undergone preclinical phase 1 or 2 tests, it's likely a good thing and that it'll save us time and be able and enabling us to provide this to patients sooner. And so there's one molecule I've kind of become fascinated with since I've joined. We can thank my former lab mate, Tom Graham, for that. But this is a molecule developed by GlaxoSmithKline, originally to treat alcohol use disorder and obesity. And it checks a lot of our boxes in that it's potent for the mu-opioid receptor, it's single-digit nanomolar. It's somewhat selective, 20 and 30-fold over other opioid receptors. And indeed, it does behave as an inverse agonist or non-agonist, depending on what paper you're reading or how you run the assay. It's probably more of an apt description. So I alluded to, I'm a chemist. This is what I do, is I develop structure-activity relationships. So here we have a somewhat palatable representation of that. I tried to keep it easy and not too overwhelming. But essentially what we're going to do is we're going to add things in a modular manner to enable us to build a large drug library or drug-like library, with the goal of exploring some chemical space around our lead compound. So the lead compound that we identified is this GSK 152-1498. Got that number memorized more than I do my own social security number at this point. And what we see is that compared to the literature, we have a comparable affinity. So this is a binding, competition binding experiment Aladdin Riyad and I did. Aladdin's a former postdoc on the T32, still in our group, doing well. And what we found is that there is a slight right-fold shift in affinity. It's not unheard of when we test these things. Sometimes if the cells are fresh, we get one value. This was repeated in triplicate, so we have some idea that this number that we measured is actually pretty valid. But more importantly, what we see is we get a large library of chemical space where our binding affinity seems preserved, right? This is still potent for the mu-opioid receptor. And what we do is once we identify hits, we send them down to the PDSP down at UNC Chapel Hill. That's Brian Roth's lab, the psychoactive drug screening program, and they're able to test for things like the intrinsic activity. So they have it normalized here versus Damgo, which is a known mu-opioid receptor agonist. And we can see virtually we have no activity from our lead compound, compound 1A. Oops. Cool. So the next thing I did was to perform what's known as an expanded SAR. The reason why will become apparent as I kind of switch gears and talk about PET labeling or radio labeling for positron emission tomography. But we can see we still are able to introduce, in this case, I've put a methyl group here, which doesn't have a dramatic influence on the binding activity. And here is a finding curve provided from the PDSP, which they kind of run a comparator. Here we can see it's about equipotent to morphine. So I'll drive home the point that we're in a pretty good area of chemical space. We've still got decent potency. And so some questions that, right, that checks our potency box, that checks our intrinsic activity box. The next question you might ask is, well, do these things get into brain? And that's a fair question if we're going to try and solve opioid overdose. One tool we use in the MET group prospectively is this boiled egg plot. And you can envision why it's called that, kind of looks like an egg. But what we're going to do is we're going to measure on the x-axis, the topological polar surface area, fancy word for how big the molecule is. The y-axis, we have the W log P, or essentially how lipophilic or not the compound is. Molecules that are in the yolk are predicted to get in brain. And we validated this with a lot of our own molecules. It can actually explain a lot of why some of our PET probes either do or do not work. What should not surprise you is that I've drawn fentanyl here. It's not a molecule I've made. It's a molecule we can fortunately just enter the SMILE string code into a database, and it gives us, it gives us a position of this on the of where fentanyl appears on the chart. And it's kind of right in the middle, predicting that it would have a high brain uptake. Again, not really surprising, given that we know that fentanyl is driving the morbidity and mortality of this opioid genocide. What might surprise you, though, is that naloxone is actually yolk adjacent, kind of on the other side of the yolk. And really, when you look at it, it's not actually in the yolk. So to me, it's surprising that this molecule is still used to displace opioids. One of the recent studies that came out by Nora Volkov's group is that in support of the FDA's decision to approve high-dose naloxone called Cloxido, it's a 7 milligram dosage. It suggests you need more naloxone, more Narcan to be able to adequately displace these potent synthetic opioids that are plaguing our street. Yeah.
Two things. Clinical folks talk about now trucks.
Mhm.
How different is it? Show up similarly?
Yeah, it actually is very close to naloxone. And here we have this double bond. If you actually just draw a triangle here, make the cyclopropyl group, they're structurally extremely similar, but they have slightly different therapeutic indications. Narcan or naloxone, good for reversing opioid overdose. Naltrexone is more used in maintenance therapy. What's that? Yeah, much longer, longer acting. So one of the key points I'd like to make here is that SAR, or the structure activity relationships I've done, enable us to, I call them play tricks. We can kind of start with our lead compound here, which is kind of on the edge of the yolk, and by changing either the functional groups, so I can take this as a triazole group, I can replace that with a benzamide, that decreases the lipophilicity, making it more drug-like, or enabling us to predict that it might penetrate the brain a little bit more, while still retaining potency. And I could do this with lots of different molecules and in different ways. I can add or replace fluorine groups. But again, we're still in a rich chemical space where we demonstrate single-digit nanomolar affinity for the mu opioid receptor. Now, given what I've shown you, we have a prediction now that perhaps this will get into brain. Some of the molecules I've made might be able to displace potent synthetic opioids. You yourself might ask, well, how does naloxone do? Does the naloxone displace C11 carfentanil? All right, so C11 carfentanil is our PET tracer of choice for in vivo studying of mu opioid receptor availability. And then you might subsequently ask, do any of the yolk compounds that are predicted to have high uptake, do these also displace C11 carfentanil? And more importantly, or most importantly, is there a difference? So to answer these questions, we did a series of PET studies in collaboration with the PenPET Explorer group. So the study, or the picture, if you've never seen one, looks something like this. So this is one of our non-human primates. We're injecting C11 carfentanil through the, I think it's a femoral artery or tail vein. This is acquired on the PenPET Explorer, and I'd be remiss if I didn't just make it known that this is one of only 2 full body PET imaging instruments in the United States. One developed here by Joel Karp. The other is in UC Davis and was developed by Simon Cherry. They're able to, in real time, give you information about where your radio tracer is accumulating. And so just a kind of a high level overview of the experiments we've designed. We've done some baseline studies to see where our C11 carfentanil goes. We've done a blocking study with naloxone. And this allows you to generate things like dose occupancy curves, which are useful in predicting how long your antagonist will stick around and engage to the receptor. And then we've subsequently done some displacement studies. We've done so at 10 minutes. That's about the time when C11 carfentanil reaches its peak uptake. And we've also prolonged it until 40 minutes. And at the 40-minute time point, we've actually done different studies where we've either administered naloxone or the GSK compound. So first, I'll just mention that our studies with naloxone showed that, yes, it does get into brain, which should be reassuring, and that it does displace opioids from the mu-opioid receptor. What might surprise you, and quite frankly, it shocks me a little bit, is that it takes about 4 to 7 minutes for naloxone to displace C11 carfentanil. I'd argue that that's a really long time for someone to hold their breath. So if you encounter someone on the street who is suffering from opioid overdose, maybe they've stopped breathing. I think this lends credence to the fact we can do better, and we should try to do better in designing new antidotes. And that it takes a full 80 minutes for naloxone to displace C11 carfentanil, such that our SUVR, standardized uptake value ratio, is equal to 1. In other words, this means that your all the binding you see is roughly non-specific binding. So
Is the route of administration also limits or?
this is for the naloxone, it's intramuscular, as is the GSK compound, which is sometimes different than what you might give on the street. So I know for if you go to your pharmacy, they'll give you an intranasal spray you can give, but a first responder might have access to something you could administer intramuscular. Yeah, it certainly might be better.
Yeah. I think the intranasal, people talk about, when we used to do naloxone reversal by injection, and it feels faster to them, and when they do intranasal, they have to tell them to wait and don't put any extra naloxone on board. Wait at least 3 to 4 minutes because it takes 20 minutes, and it is agonizing, right? Because you want to do something extra, and so
Yep.
people, they can be initially given too much naloxone. So that's a tricky thing too. So basically, we need better.
It's a delicate dance, right? You don't want to give someone too much at first and then bring about withdrawal. So I think there is still much to be learned from these studies, and I think subsequent studies might address route of administration and what gets to the brain quicker.
By the way, when people get reversed, it's a very sort of alarming experience for them. They're brought back, but they're very disoriented, they can be very agitated, and not in the mood to sign up. Send treatment back. In fact, because they're feeling such precipitated withdrawal, they're going to back on and get some hope. And so it's a very alarming thing, and all of you seen the videos at Kensington and Convention Point, where they're doing syringes and doing reversals, sometimes more than 1 time per day per person, because there's this cycle. So it's a mess, and us is going to fix it.
I hope so. Wow, no pressure. Okay. Subsequently, we asked the question, does our lead compound get to brain? And so we did similar displacement studies. So we're still only administering our naloxone at about 10 minutes, but we've done some subsequent injections where we've explored now administering the GSK lead compound at about 40 minutes. We could do kind of an apples to apples comparison of how fast naloxone displaces carfentanil and how fast the GSK compound does. Based on this data, it suggests there's not much of a difference, although I would still argue that I think it remains unclear and that we'll need a couple more studies. I think exploring routes of administration may help us answer this. And one thing I'm keen to investigate moving forward is that I'd like to kind of repeat some of the blocking studies where you administer the cold antagonist beforehand and then can generate really nice dose occupancy curves to see how long the antagonist will quote unquote stick around and maybe preclude rebinding of the opioid from the mu-opioid receptor. Another thing I'm keen to investigate, and this is something I'll need to collaborate with someone on, is studying if these exhibit respiratory rescue, right? So maybe inducing respiratory distress in a mouse, seeing what naloxone does, and then seeing what the lead compound does. I think those are both pretty meritorious ideas that I hope as I transition away from synthesis, I can now try and have conversations with more biologists. But we're not out of the synthetic woods yet, don't worry, because I'll switch gears now and talk about how I've used my background as a synthetic chemist to start to develop PET tracers, positron emission tomography tracers for the mu-opioid receptor, and the key challenges that I've been trying to address in that. So as I mentioned, C11 carfentanil is like our bread and butter for imaging the mu opioid receptor. It works very well, but there are a couple issues with its use. One is that it can actually cause respiratory depression even at tracer doses. So you give nanomoles of this to a patient or non-human primate, you could still induce unto them respiratory depression. And it also requires a Schedule II DEA license to be deployed clinically, which I would argue has precluded its widespread availability. So in transitioning from developing therapeutics to now PET tracers, there's a few different criteria we might want to be concerned with. One is this binding potential requisite, and that is the Bmax, or the receptor concentration for the mu opioid receptor, isn't all that abundant. So in order to make sure you get decent signal to noise, you need to have a Bmax KD ratio that's generally above 10. And that kind of constrains our KD, or a binding affinity, to being single digit nanomolar at most, right? We ideally want something a little bit more potent. And also something called the shape flask log D. This is useful for predicting non-specific binding. Given the kind of rightward shift towards more non-potency that we've seen with my lead compound, I would argue that we should be okay with a compound like 4A. It should exhibit sufficient potency. It's a non-agonist, so this will get us around hopefully any regulatory requirements, and stop us from accidentally sending a patient or non-human primate into respiratory depression during a somewhat routine medical exam. So some questions we might then pose are, can methodology be developed to radiolabel some of our best therapeutic candidates, and will these meet the criteria that I've just mentioned? And so I kind of just want to give a brief overview, and chemistry might be a little bit different than what you're used to, and I'll try and keep this as quick and painless as possible. But there are key differences between benchtop synthesis, something we can do in a lab, and something that you do in a cyclotron. The biggest constraint is that the rule of thumb is you want to complete your radiosynthesis by about 2 half-lives of your radionuclide. So for carbon-11, you've got 40 minutes to get your synthesis done, your product purified, rushing out the door to QC, and simultaneously bringing it to a patient. The other issue is that you generally don't want to inject a lot of this material, especially if it's an agonist. So this necessitates dilute reaction conditions. We do this on very small scale. This can slow down a reaction. So some things we do are increase the temperature. So the rate will double about every 10 degrees. You bump that temperature up, and you can do other things like high catalyst loading to help you get the reaction cooking, as it were. So the synthetic route I designed was to take this aryl bromide, and what we're going to do is we're going to perform a cyanation with the catalyst. Our source of radionuclide is going to be C11HCN. Yes, you read that right, HCN. And then we're going to hydrolyze it to the corresponding benzimide. We're going to try to get this all done in 20 minutes from end to barbitide. So that's once the radioactivity has reached your hot cell. This isn't an exhaustive list of all the conditions I've tried, but these are 5 of the most interesting. And what we see is we do actually get conversion, and it is contingent upon your choice of catalyst. Other decisions went into this, such as reaction time, temperature, solvent choice. All of these things affect your ability to affect this synthetic transformation. But we do see we can get 50 or 60% conversion, which is okay for transitioning this from benchtop to the hot cell in the cyclotron. And I've actually tried this a few times in the cyclotron. I will mention there are some ongoing optimization issues that I'm working on as it relates to catalyst solubility. I think I've got it figured out. If this meeting were a week later, I might have some different news for you. But right now we're 0 for 2 on the radiolabeling efforts, but I'm pretty confident we'll figure this out. And if not, there's actually a different way in which we might do this. So I'm just sharing this methodology because it was developed at Penn in collaboration with Merck and Princeton, in this photo redox methodology, in which we take C11 methyl iodide, and we can couple this to another aryl bromide using iridium, nickel, and light. Enables you to actually radiolabel Celebrex, and this is something I've done in the cyclotron. So I know this methodology is pretty robust and it works. And so in the coming weeks, our cyclotron is actually down right now getting maintenance, but next week I plan to be back and trying to get similar methodology worked out for the radial labeling of 13A, which is a precursor that I've made, I have on hand, in an effort to radial label it into carbon 11.7A. So just to conclude, I think we're in a really nice area of chemical space. My short-term goal would be to optimize the labeling conditions. I found something that works. It's just a matter of, I think, optimizing solubility conditions for the catalyst. And my longer-term goal would be to validate this tracer and hopefully, and I don't like to make promises, but especially in research, but maybe to have this validated for some in vivo studies by the next base day. We hope.
Very good. Thanks.
Sure. And then the most important slide last, I'm sorry. Just to give a shout out to my boss, Bob, who's been great at kind of forming an environment where biologists, chemists, and psychiatrists can all kind of come together and solve interesting issues. CJ is a wizard. She's a researcher in her lab who does a lot of PET imaging. She's a lot of the PET graphs you saw were from her. Olad and Riyadh, who helped me develop some of the in vitro binding assays, the cyclotron crew, Sharon and Alex, and then the P30 personnel, Anna Rose and Julie, thank you. And I'd be happy to entertain any questions, comments, or bitter remarks.
Back to our whole chemistry days.
We have one question.
Yeah. But you're just. I think this all your labeling, that was all the.
Yes.
Your all the goals like this.
Yeah, the nice thing about the PET Explorer is you can actually get not only full body, but you can really get a good idea of every region of the brain. So I do have some different PET images here that focus on almost every region of the brain you might be interested in, not just the central nervous system, but also the periphery. We chose thalamus because we know it's a mu-opioid receptor rich region. We compare it to the cerebellum in non-human primates, occipital in human primates, because those are areas that are generally devoid of any mu-opioid receptor binding.
You might want to talk about just for fun, since people are doing animals and about humans and primates and others for reference reason.
Yeah, so the CJ from our group actually recently presented at SNMMI that there are differences, species differences in, let's say, non-human primates and rats versus humans. So traditionally, I think the occipital has been used as a reference region for almost all three of them. But what CJ found is that it actually should not be the case. I don't know if I have the exact, I don't have the exact slide, but she showed or found that there was some displaceable binding in the occipital in our non-human primate model. And so she made a pretty compelling argument for rethinking that and then changing a reference region to the cerebellum.
So reference meaning an area devoid of movement.
Correct,
Yeah.
yes, yeah.
So the cross species translation, people always, worry about, well, be careful, there are differences and it is something that being aware of it, having the data in all three species, very, very useful. So I just thought I'd mention that. The other
Thank
thing is, it's really understated to say that carfentanil is challenging. I mean, it's a difficult place to work with, right? Because small frame, people who are smaller can get these pharmacologic effects of tracer doses. You don't want that. And you also don't want to have to keep naloxone, right beside them when you're doing the science. Or if you're going to start feeling dry. I mean, that's not ideal. That's not ideal. Right, so it's what we've had, but it's why we need to process it.
Yeah, no, I was one of those people with Karen Lerman's
Were you?
smoking study.
You got some
I got
pharmacologic effects.
I got
And
carfentanil.
you're not even especially petite, right?
I'm not actually petite. Thank you, Anna Rose.
You're a good average sized person, but they talked about it. I never knew this study directly, right? But they made it sound like if you were a fragile, 5 foot and under female, that maybe would be more vulnerable. The big boys, or even average people, no problems. Now we know.
Now we know. This is perfect. Oh, Greg, thank you for jumping up there.
Not a problem. Can everyone hear me all right?
Just like to say that Julie's the perfect size.
I think we're good in terms of All right, I'll put it up as loud as I can. Go ahead.
Okie dokie. Well, thank you very much. Nice to be here today. Sorry, well, I can't be here today in person. Sorry about that, everyone, but I'm happy to be here to talk about the research that I've been doing for the past year or so in Dr. Corder's group, and with the aid of the T32. So as my title kind of shows everyone, what we've been doing is kind of coming up the question of how to tackle the opioid-based pandemic and how to treat and study the opioidergic circuits in the brain from a slightly different angle than some of the projects we've just heard from a little bit recently, where it's opposed to trying to develop new pharmacologies, we're actually trying to look at this from a gene therapy-like perspective for our tool development kind of work that I've been heading up in Greg Corder's research group. So as everyone has kind of said, as
Okay, we were just gonna, do you think you want to take your mask off? Can you?
Are
Sure.
you at home? Yeah, okay, thanks.
sure, is that a little bit better?
Yeah, it is, thanks.
Okay, great. All right, sorry. So just a quick introduction here, as you've all heard from everyone else who's given all their lovely presentations thus far. Obviously, a particular interest to us and to this research group in general who are on the T32 is the question of opioids and the opioid addiction-based problem that is hugely problematic both in the US and the world at large. Obviously, we want to find better ways to treat people that avoid the kind of overdose potential that's associated with opioids, but another issue that comes with finding the best way to treat people by targeting the opioidergic system is that we also don't want to completely mute the inherent benefits that the opioidergic system and pain processing has in our day-to-day lives. So how do you kind of thread this needle of trying to find better and new novel treatments that allow you to target these pain processing cells and manipulate them in ways that do not completely subvert the physiological function these circuits and these cells perform, but also don't push it into the extreme where you have the issue for dependence that arises with a lot of the typical pharmaceuticals that exist for treating chronic pain conditions and other maladies of the opioid system today. So that's kind of where some of the work that has been done and discussed by a number of the other excellent trainees so far with using new tools to selectively identify and manipulate opioidergic cells and circuits within the preclinical space. We're actually interested in seeing whether or not this is something that can be used in a practical sense to kind of jump the gap from the bench, so to say, into the clinic. And we've been working for the past couple of years or so to develop these new novel tools that utilize viral vectors to see if we can selectively target these cell populations of interest that a lot of us have been recently talking about today. So kind of jumping right in, the premise of how we've been going about this is by trying to utilize adeno-associated viral vectors, or AAVs, that again a number of other trainees have talked about, to selectively target opioidergic-based cells and circuits within the brain. Again, as some people have alluded to, these viruses are somewhat in vogue right now. They've been very heavily used, especially in the preclinical space, and are starting to make waves in the clinical space with a lot of new gene therapy-like companies, and even our own GTP center here at Penn, developing a number of these vectors that can selectively target certain populations of cells and insert a number of different transgenic-based tools into these cells to either manipulate or change DNA at a cellular level. The AAVs are particularly interesting, mainly because they have a relatively low immunogenicity. They also have the ability to store a pretty large amount of genetic information in them, and they've been shown to have pretty decent tolerance for targeting different parts of the brain as well. So here, this is just a quick outline of how our group, in collaboration with Carl Deiseroth and his biological core director, Charu Ramakrishnan, to develop viruses that are actually able to target the promoter region of the mu-opioid receptor gene. And here you could see on the top of this first cartoon, we're showing how we went around to design a couple of different constructs that were able to selectively target this promoter region within the mouse mu-opioid receptor gene, OPRM1. And we've also done this for developing a kind of virus that can selectively target this same region in the cognate human OPRM1 gene as well. And this is just a quick outline again of how we went about developing this. And we eventually ended up settling on this first construct that you can see, this topmost green bar cartoon here, which was able to selectively target a pretty decent region of the promoter that allowed us to test it in a number of different functional ways. That's kind of outlined here on the right of this cartoon, where we're showing the different kinds of constructs we were able to make. and they have a number of different transgenic cargo that can be inserted into them that a couple of the other previous trainees have talked about in pursuit of their own research. I'll kind of go into a little bit more depth in terms of how we utilize these different kinds of vectors to start to functionally and selectively validate the utility of our virus in vivo in animal models and then in higher order mammalian models as well. So here, this is kind of some of our first round of work that we've been doing with these viruses, headed by a number of folks in our lab, including myself, with direct injections of these viruses via intracranial injection techniques into a couple of different candidate regions of the mouse brain that are known to have high expression of the mu-opioid receptor. In this case, we're looking at the medial prefrontal cortex, which is known to have a number of different opioidergic populations within a couple of these different regions that you can see outlined in our cartoon here. We were able to show that this virus expresses very well within these regions and seems to be selectively restricted primarily to neurons, as you can see via this NUN neural stain right here, while sparing other surrounding support cells within the brain. Now, specifically here, we're staining for microglia. You can see in this overlaid image here on the bottom right that we don't really see a lot of transfection occurring with our virus in these glial support cells, but mostly primarily in the NUN targeted neurons. Moving on from here, we've done a couple more studies since we last kind of spoke and updated this group to more selectively validate the expression of this virus in only cells that should express the OPRM1 gene in mice. Here we used a combination of in situ hybridization studies as well as antibody-based staining to show that what we appear to be seeing when we look for the overall expression of our virus, which in these pictures is tagged with a EYFP fluorescent tag or reporter that can show us where they are in slices when we take them and look at them under the microscope. And we're also able to stain for this reporter with RNA-based probes using this in situ hybridization-based technique. And we can also stain for a couple other proteins of interest here. In this case, we're looking for the mu-opioid receptor itself, which we can stain with an antibody for. And we can also see how this overlaps with the expression of both an EYFP and an M-Cherry reporter when using a couple different variants of the virus that I mentioned before. Specifically here, this is showing us examining another region of interest. This is the central amygdala, which, as Adrian and a couple other people have mentioned, is also known to have a high expression of the OPRM1 gene and more positive neurons as well. And we see that at both of these levels, there is good overlap with our virus, with both the transcript reporter for this OPRM1 gene of interest or mRNA of interest. And we also see that it seems to overlap well with the signal that we see for the mu-opioid receptor protein, as well as when we utilize Cree lines that are selectively able to label from birth the expression of OPRM1 within these regions. We also see good overlap with our virus in those areas as well. We've gone on to also do a little bit more extensive validation and quantification of this across a number of different regions. A couple other folks again have mentioned today, specifically looking in the central amygdala, the dorsomedial striatum, the ventral tegmental area, and the medial prefrontal cortex. Again, all candidate regions that are known to have high expression of OPRM1 and or the more protein within the mouse brain. We quantify this across a couple of different animals, specifically, as some folks have mentioned, using these Cree recombinase-based reporter lines that have the expression of this Cree recombinase protein directed by the OPRM1 gene, using both a commercially available line developed by Richard Palmer's group and Dr. Blendy's own in-house developed mouse line that she generously provided to us. And what we were able to see is that within all these regions across these two animals, we see pretty high overlap with the reporter fluorophore that's tagging our virus with the Cree stain that we're actually able to use with an antibody-based technique to show that there's very, very high overlap between these putative OPRM1 positive cells of interest and our virus in terms of which cells they're selectively transfecting as opposed to surrounding cells that shouldn't have OPRM1 in them. So we wanted to show that these tools are actually very useful for a number of different groups. Again, obviously nothing is done only in mouse models in the preclinical world, despite the fact that there's a lot of transgenic animals that are useful for doing that, as we've mentioned with these OPRM1 Cree-based lines. But a lot of people do important research in the opioid space, both at the level of addiction and pain using rats and shrews and other model systems. However, they don't have the wealth of transgenic animals that the mouse researcher world does tend to be pretty blessed with. So what we did was working in collaboration with Dr. Matthew Hayes's group here within the TRL. His postdoc, Tito Borner, was able to help us out by doing some injections in rats and shrews for us. And what we're able to see is when looking at complementary regions, at least across the mouse and the rat, in this case, looking at the central amygdala and the VTA again, we see very nice restricted and selective expression of our viruses within these areas of interest, targeting again more exclusively the neurons and none of these glial-based populations. And we're looking in the shrew in the area known as the nucleus solidaris tract, the ATS, an area that's known to be involved in the emetus response in shrew models that are given opioid overdoses. So studying this kind of vomit-like effect that happens when animals are given a noxious amount of opioids, we're actually able to see pretty selective expression of our virus to this region of interest as well, proving the utility of this virus, hopefully across a number of different smaller mammalian models. From here, we also wanted to show that this virus and the different variants of the virus are useful in functional-based tests. So obviously outside of just labeling for anatomical studies' sake, we want to show that we can use this in a way to meaningfully manipulate pain and noxious nociceptive-based behavior in these mice, as well as opioid-seeking based behavior in these animals, to see if these viruses are useful to the maximal number of preclinical researchers who might be utilizing them, especially mice. So here in this study, what we did was we utilized one of our DREAD-based viruses, again, DREAD-based methodology or chemogenetic-based methodologies were explained pretty well by some of the past trainees. But again, it's a desire receptor activated exclusively by a drug that should not have any other cognate receptor present in the system outside of those that were transfected with this virus. So Dr. Corder and Sophie Rogers, another member of our lab, injected these mice intrathecally with this virus between the L3 and L5 lumbar region of the spinal cord with an eye of targeting hopefully the dorsal horn region of the spine with this virus. As we could see in these images and some generic quantification that was done, we do get pretty nice restriction and transfection of our virus within this region of the spinal cord in a number of different cell populations that are known to respond and transmit stimuli and information related to nociceptive-based stimuli from the periphery up into the actual CNS. And following a couple of manipulations in these animals, again, kind of building off of what some of the past researchers have shown you in terms of how to measure different nocifensive-like behaviors in these mice in response to noxious stimuli, using both a mechanical-based stimuli, a heat-based stimuli, and an inflammation-based stimuli with a formalin injection in the high paw. What we're able to show is that when these animals were administered our virus in the spinal cord and then given in a dose of the drug CNO, which should selectively activate this receptor, in this case, we are using a DREAD vancomycin inhibitory receptor, we see both a increase in the pain mechanical threshold. So these animals are less sensitive to noxious mechanical-like stimuli and a reduced latency to their overall response to thermal-based manipulation. So again, they are less sensitive to heat-based or noxious heat-based stimuli, which seems to trend with a reduced, with an overall reduction in their pain-based responses. Similarly, when looking at this formalin-based task, we also see that these animals show a reduced nocifensive-like behavioral profile when compared to animals that were not administered the virus or the CNO compound, kind of giving us an overall sense that these animals seem to have been selectively targeted within this OPRM1 specific population, which a lot of literature has shown that manipulation in this region of the spinal cord using other kinds of opioidergic-based compounds or pharmaceuticals with either a cannulation or something of that nature or a systemic-based injection can reduce the overall activities of these cells. We seem to show that our virus can replicate these similar effects when selectively targeting these neurons within the spinal cord. Moving from here, we did a couple other functional-based assays to more further prove the utility of these viruses, again, in the preclinical space in mice. As some other folks have mentioned, there's a couple newer, more in vogue techniques coming out nowadays in the mouse behavioral research area that allows you to do imaging of in vivo calcium-based signaling within the brain using a couple of different in vivo-based techniques. This is a technique known as in vivo fiber photometry, which makes use of a GECI, which is known as a genetically encoded calcium indicator that allows you to see the change in overall calcium-mediated based activity via the change in the overall fluorescence of the construct that has been injected into the area based on a FRET-based response that we can now measure using a fiber optic cable that can be implanted over the area that the injection was administered. So here we kind of give a brief outline of what we did with these animals with injecting our opioid-based promoter targeting virus into, again, in this case, the CEA, which is an area that, again, is shown to have responsive-like effects when nociceptive behaviors are studied in these mice. We're able to see nice expression overall and selective restriction to this region of the brain when administered. And we then tried to run these mice through a battery of different kinds of basic behaviors to see overall their offense to acute noxious stimuli. And then we went ahead and tried to see if we could measure different kinds of responses in these animals when they were presented with a morphine-based drinking paradigm. So in our acute-based studies, what we're able to see here, these graphs are readouts of the overall gross change in calcium-mediated fluorescence signal. This delta F over F is essentially the overall change in the baseline or background fluorescence compared to a specific event that we were able to time lock our recordings to. So this increase that you're seeing, or these spikes in the graph, are when we were measuring the overall response of the animal to, in this case, in our far left graph on the top here, a air puff startle response that we applied to the animal under its enclosure, in which you can see that there was a large increase in the overall fluorescent signal that was occurring specifically in response to this air puff that we were only able to see in the mice that got the GCAMP-based version of this virus, not just the reporter virus alone. We then went on to show that the same kind of effect could occur in these animals when we applied a hot water or a noxious thermal stimuli to the left hind paw of these animals, with again, the GCAMP group showing a nice increase in the overall calcium signature when compared to the control group, in which really nothing was able to be observed. From here, we also wanted to see if this kind of behavior was directly affected by the administration of morphine. Again, showing that hopefully we selectively targeted these OPR1 positive cells, which should be sensitive to morphine-based manipulation. And what we do show is that in these same animals, when they are given morphine dose acutely right before this hot water hindpaw stim, we do see a significant decrease in the overall peak amplitude and the area under the curve of this calcium-based response, indicating that there seems to be some modulation of that cell population's response when the morphine is on board. Lastly, with these animals, what we also ended up doing, as I mentioned before, was a forced morphine drinking paradigm. This was again to get at the question of whether or not these tools could be useful for people in the opioid addiction-based space as well. And what we show here is that we put these animals on escalating forced drinking regimen, going from 0.3 mg per ml to 0.5 over the course of 7 days, after which the animals were forced to go through precipitated withdrawal via an IP injection of naloxone. Following this, we gave the animals a brief recovery period after they were injected with the naloxone. And then we put them into a clear plastic enclosure where we could score and observe their overall withdrawal-based behaviors following the acute naloxone injection. As you can see again from this graph on the left, when compared to the animals that were only given a controlled substance, in this case, a saccharin or a sweet substance that was spiking their drinking water, the morphine drinking animals showed a pretty remarkably noticeable increase in the overall number of calcium-mediated events that were present following the administration of this drug, whereas the animals that were not forced to drink morphine did not really show much perturbation following the administration of naloxone, which seems to correlate with some of the behaviors that we observed in these mice. They're not presented here because we're still scoring them, but overall, this seemed to indicate that using this GCAMP-based variant of our virus, we could actually measure in real time changes in the overall neural activity that's associated with withdrawal-like effects in these animals, and that these effects can also be multiplex and time locked to measuring specific behavioral specific behaviors that's typically associated with precipitated withdrawal in rodents. Now, for the last couple of things that we just wanted to go through in terms of showing the versatility of our viruses, utilizing a intersect-based system, which allows us to inject one virus in one region of the brain, and then another virus in a different area of the brain, and the expression of both is dependent on them to interact in a way in which something like a Cre recombinase or another essential part of these viral expression systems is dependent on the encoding of one virus and the other. We're able to show that we can target selective populations of opioidergic cells within different regions of the brain. In this case, on the far left top here, we're showing that we can use another promoter-based virus to target GABAergic cells within the cortex selectively in conjunction with our mu-opioid-based promoter-based viruses to, again, target a specific subpopulation of these GABAergic mu-opioid positive cells within the MPFC and in somatosensory region 1. And we also show that this can also be used when multiplexing with viral constructs that depend on both the expression of a Cre and a flip-based recombinase, as seen in these two pictures here. Below, we're also showing that we're starting to explore using these viruses in a way that will allow us to multiplex them and utilize them with different kinds of capsids that allow for transfection and targeting to both the CNS and the PNS directly. And the last two things I'll just quickly talk about is we finally want to take these viruses to the next level and show that they can have some kind of utility in non-human primate and animal models that are more directly associated with translational based studies, kind of a step away from humans. And what we show here is that using the aid of our collaborators in Michael Platt's group, when injecting these viruses into a couple of different regions in the macaque, we can actually get pretty decent expression and selective transfection of these viruses, specifically in opioidergic based cells within a few different areas of the macaque brain. And lastly, using this work, using collaboration with our friends over at Brigham and Women's, we're actually also able to show that these viruses seem to be useful in vitro spaces for targeting both nociceptors isolated from human iPSCs, as well as selective dorsal root ganglia neurons that can be isolated from the mouse, showing that they might have some versatility in high throughput based screens. So I'm sorry if I'm a little over time here. I'll just wrap this up. This was just on another project that I'm doing in the lab. So I'll just end by again thanking everyone in the group, particularly obviously Dr. Corder, my mentor, and a number of other folks here who are in bold, who were particularly instrumental to the completion of this work. We currently have this in submission at NatCom, so we're going to keep our fingers crossed and see what happens. But again, thank you everyone for your time and everyone for the funding too. And if I do have a minute, I'll answer any questions.
Great, thank you. Can you hear our clapping? Is there any last minute first question before we move on? I think they just want to drink wine now, Greg, so.
Sure, that's fine.
Thank you so much for that presentation. We miss you and hopefully we'll see you soon and feel better.
And we will eat your
Sure.
slice.
Yes. So I guess you need to stop sharing. Great, thanks. Okay, we're still here. For who's Cindy's next, right? Yeah. All right, let me find Cindy. Here we go. And then we will share screen, share. Perfect. Okay.
Just to brag on Cindy really briefly, she's had a nice paper already publishing with the renowned Marin Weirs, but also the director of NIDA, Dr. Wolkoff. She published a paper, first author in this past months, and she also received a CHPS Junior Investigator Preliminary Feasibility Grant from Penn, and congratulations, Cindy.
Thank you everyone for coming to this talk. I'm going to be shifting to more clinical research and to share some of our ongoing research on the role of nutrition in recovery from substance use disorder, focusing specifically on opioid and alcohol use disorder. So the role of nutrition in substance use disorder is often overlooked, and I hope to convince you guys that there is need to be consideration for nutrition in the treatment of substance use disorder. So opioid use disorder is strongly associated with malnutrition. This population, they're not the most financially stable. There's high rates of homelessness, and also the cost of obtaining drugs is very costly. So they often don't have the financial resources to obtain high quality nutrient-dense food. Also, chronic use of opioids attenuates reward processing. So they don't really get that pleasure that you and I get from food. And also they tend to prefer foods that are high in fat and sugar, which don't always have the best nutrients. And also chronic opioid use causes GI distress that decrease the amount of nutrients that's being absorbed, and also it causes oral decays that further prevents food consumption. So one study compared opioid in OUD individuals from participants in a clinical trial network over at NIDA, and they compare that to the general populations from the NIH surveys, and they show that, if you looked at it, the opioid users, they show significantly decreased BMI as well as lower prevalence of obesity. So switching gears a little to alcohol use disorder. So people with alcohol use disorder, they also show malabsorption of essential nutrients. The most notable example is thiamine deficiency, which causes Wernicke encephalopathy, which is characterized by motor impairments, altered mental state, and ocular abnormalities. And it is now common clinical practice to supplement with the vitamin thiamine for the treatment of alcohol use disorder. From a nutritional standpoint, ethanol is really interesting because it is the only substance of abuse that has a caloric content. It has 7 kilocalories per gram. In comparison, carbs and protein have 4 and fat has 9. So it's a substantial amount of calories. And individuals that consume alcohol, because of the extra calories, that translates to increased body mass, kind of like the beer belly that some people have. However, what's really interesting is that in people that are often use high doses of alcohol, they actually show lower BMI and lower fat mass despite the increase in alcohol and calorie intake. But that is just for more of the high alcohol consumers in AUD. So there is the thinking that there's altered metabolism associated with chronic use of alcohol, namely increased metabolism and increased fat utilization. Also, alcohol has been shown to decrease muscle protein synthesis, and that contributes to myopathy in some individuals with alcohol use disorder. So given what kind of shared about substance use disorder and nutrition, we really want to get to look into what is the implication of nutritional status on recovery from substance use disorder, and is there a role for nutritional intervention in aiding recovery in individuals with substance use disorder. So I'm going to share some data from our ongoing project. One is looking at the role of BMI as a predictor of naltrexone treatment retention in individuals with opioid use disorder. And the other one is ketone ester supplementation on alcohol responses and consumption in individuals with alcohol use disorder. So starting with our first study on BMI and opioid use disorder. So there is increasing research looking at this intersection between food intake and use of drugs of abuse. And it is postulated that these drugs of abuse, the reinforcing effects of these drugs of abuse act on the same neural circuitries that reinforces survival behaviors such as food intake. So I thought maybe the lack of the deficiency of energy and nutrients in this population may drive consummatory behaviors that translate to craving and exacerbate opioid seeking. So in the study on the left, it was a study in which rats were trained to self-administer heroin And then they underwent withdrawal, during which some of the rats were food restricted and other were fed ad libitum. And then the heroin seeking activity is tested. As you can see by the black dots, those are the food restricted rats. And these show an increase in heroin seeking behavior. And then in the study on the right, these were done in mice, in which mice were made vitamin D deficient, either using a transgenic knockout of vitamin D receptor, or they were made vitamin D deficient using a dietary intervention. And as shown here, the vitamin D deficient mice showed increased reward sensitivity to morphine. So we use BMI as a proxy for nutritional status and examine that on predicting naltrexone treatment retentions in individuals with OUD. This is a retrospective secondary analysis of 2 intervention study in OUD individuals that were receiving monthly injections of naltrexone. So these patients were offered up to 3 injections of naltrexone. We categorize completers as those that receive all 3 naltrexone injections, and non-completers as those that are missing 1 or 2 injections. So we did a binary logistic regression looking at individual variables in predicting treatment completion. As you can see in red, BMI, high BMI was positively associated with increased likelihood of treatment completion. In consultation with our statistician, we, in our multivariable model, we decided to include any variables that showed a p-value below 0.25. In our multivariate analysis, BMI was still positively correlated with treatment completion, even after adjusting for age and opioid craving. And also opioid craving was also significantly associated with treatment completion. Moving on to our next study, looking at the role of ketone ester supplementation on alcohol responses and consumption. As I kind of alluded to previously, alcohol causes a shift in energy metabolism, and that's also seen in the brain, where alcohol has been shown to decrease glucose and increase acetate utilization. And this effect is accentuated in individuals with AUD. It is thought that brain energy deficiency during detoxification, when acetate levels are decreased due to a lack of alcohol, may contribute to withdrawal symptoms. A study published last year by my mentor, Dr. Colonel Years, showed that a ketogenic diet was efficient in reducing the need for benzodiazepine for the treatment of alcohol withdrawal, and also reduce craving for alcohol. But ketogenic diet is a very extreme diet, and it's really hard to maintain. And as an alternative, there is a ketone ester supplement that also provides ketones. This is some preliminary data. This is only in 5 individuals, in which individuals were brought in once at baseline and then after consumption of ketone ester. And you can see that the ketone ester reduces glucose metabolism, and that's kind of consistent in the shift with energy metabolism. And then we want to see whether ketone ester is able to reduce alcohol consumption and craving in individuals with alcohol use disorder. So in this bar lab, we recruited individuals with alcohol use disorder. This is a crossover design in which they were brought in once with placebo and then another time with a ketone ester. I'm going to be sharing some interim data. This is only in 5 individuals. We kind of just unblinded our first 5 individuals last week. So I'm going to be sharing some of our data. We also collected MRI, but that's not been analyzed. So these individuals were asked to complete some questionnaires, the AUQ, the alcohol urge questionnaire, to kind of look at craving, and also the BAES and the DUQ to assess their responses to alcohol, as well as we measure multiple breath alcohol concentrations. And as a part of the bar lab, we were first given a alcohol priming drink, and then we participated in a bar lab in which we were given 4 alcoholic drinks. Each one is worth $3, and they can either pay for it or drink, or they can keep that $3. So that is that was done once here and then another times here.
I was gonna say the wine's free at the end of this.
Yes.
Just in case you're worried, but
So we first measured breath alcohol concentration, and there seems to be, it's not significant, there seems to be an effect of ketone ester in increasing alcohol metabolism and decreasing the breath alcohol concentration. But as I kind of talked about in our previous slide, they were participating in this bar lab. So when they choose to consume the alcohol kind of varies, but then the time that we measure the breath alcohol concentration, that kind of stay the same. So then, so that may have confounded some of our BRAC data. So we also did a pairwise t-test to look at the breath alcohol level right after the priming drink, which everyone got the same amount at the same time. And then there seemed to be an effect of ketone ester in increasing alcohol metabolism and decreasing breath alcohol concentration, which was really surprising because I actually hypothesized the opposite direction, and that it would cause increased intoxication. But we're trying to still figure out why that is. We also measured some subjective levels of intoxication, and there doesn't seem to be any effects. During the bar lab, there wasn't any significant difference in alcohol consumption, and there wasn't also any effect in alcohol craving using AUQ. So in conclusion, our first study demonstrated that increased BMI was associated with increased likelihood of completing naltrexone treatment in OUD individuals. One limitation of this study is that it is a secondary analysis, and we are limited by the availability of the data. And we use BMI as a proxy for nutritional status. That's not always the best measurement of nutritional status. Just because you have a higher BMI, you might be eating too much cookies and ice cream. That doesn't necessarily mean that you are very in a very nutrition positive state. So it'd be really interesting to look at the role of diet quality or appetite questionnaires on treatment outcomes. And also it'd be interesting to look at whether for nutrition, how does that affect opioid craving in individuals undergoing detoxification. And then with regards to the ketone ester study, our data suggested that there is an effect of ketone ester supplementation in increasing alcohol metabolism, but there's not really effect on alcohol consumption or craving, and that might be just because it was a sample size of 5. So future study, I got a small pilot IMPACT grant from Penn, where I want to look at the role of ketogenic diet and ketone ester on alcohol metabolism. I kind of want to differentiate the effects of ketones versus ketogenic diet, which causes a shift in energy utilization. And also in this study, we won't have like everyone will be drinking the alcohol around the same time, so it'd be easier to make sense of the data. With that, I just want to thank my mentors, Dr. Korean Mears and Jin Shi, as well as our lab, and thank you to our co-directors and for the T32 program for this fellowship, and I will be happy to take any questions.
Now this is one that Ari is well smiled about it. We don't know why the breath alcohol concentration is changing with the ketone ester. So you guys have probably kicked around the ideas.
Yeah, I feel like it has to be on like alcohol dehydrogenase or acetyl dehydrogenase. I think that is the most likely scenario. Also, I'm also interested in like also blood ethanol concentration, because there is one study that showed ketogenic diet may may produce false positives. Actually, no, this is, yeah, so maybe there might be discrepancy between breath and blood, but I really strongly doubt it. But yeah.
It's interesting. And it's sort of, I'm curious, there's increasing in my also.
This was only in the so I haven't really seen any data looking at BMI buprenorphine responses, but actually buprenorphine causes an increase in weight gain. So I'm not really sure how BMI would affect.
I bet when you do that, it's now, there's no data out there now. There's a free idea on
So then
along.
I think one thing that we have to be careful is BMI also may affect pharmacokinetics. Is it just because it's working differently, or is it? Not yet, but that's something I hope.
Thank you. Yes. Sorry, I'm not, I mean, you to trip you, but I am tripping.
So sorry, this is moving on. I know everybody's getting late in the day, but we have one more talk, and Kate was a pre-doc supported from my lab on the T32, and now most recently just 1 year of a postdoc because she actually just got her NRSA, her F32. Right?
Thank you.
And going to tell us about some complex multi-layer stuff.
Thank you, Julie. Yes.
Like it's 3 years. It's crazy.
It's been a while. Great. So I'm excited to tell you all about a project that's in very early stages, in which we're working on modeling the interplay between neural activity and gene expression in mice and humans. So I'm going to be talking about multi-layer networks today, which are very useful for modeling systems of components that interact in multiple ways. So for example, in this ecosystem network, at the bottom, we have a spatial layer that gives the distribution of a series of trees. And then the red and blue nodes that you see represent birds that inhabit each of the trees. And you can see in the social layer and mating layer representations of ways that the birds interact with one another. So edges in the social layer connect birds that have interacted socially. Edges in the mating layer connect birds that have mated. And these two layers together form a multiplex network because each entity, the same entities are present in each layer and make connections only to themselves. Whereas the social layer and spatial layer form an interconnected network where each bird, the edges connect each bird to the trees that it's visited. So you could imagine using a representation like this to address questions about how the spatial
location of the trees where the birds live inhabits the likelihood they interact socially and the likelihood of mating. Multi-layer networks can also be very useful for modeling biological systems like the brain. So here we're looking at two representations of the same brain. On the left-hand side is the structural connectome, where we can see the regions are connected according to the density of axonal fibers between them. And then on the right-hand side, we're looking at a functional representation of the same brain, where we can visualize correlations in neural activity between every pair of regions. So you might imagine representing these two layers of the brain as a multi-layered system, where you could then ask questions about how similarities in structural connectivity impact similarities in neural activity, or vice versa. So in this project, we're particularly interested in addressing the question of how we can drive a multi-layered system to a desired state in the case where we may have access to only one layer. And we think that addressing this question in the long run could be useful for thinking about how the brain transitions between health and disease states, where disease pathology or dysfunction may impact multiple layers. This is certainly the case for substance use disorders, for example, where we see differences in functional connectivity, both at rest and in cue reactivity paradigms, as well as we see changes in gene expression. Then building on that question, we could think about how we might target an intervention to impact pathology or dysfunction in multiple layers when we only have access to one layer. So for instance, if we were targeting the neural layer, we might think about using a stimulation-based therapeutic to either suppress or potentiate neural activity within a particular region to drive both layers to a desired state. Or if we were using a pharmaceutical manipulation, we might think about modulating the activity of a particular neurotransmitter system to affect gene expression and ultimately neural activity. So we propose to drive a multi-layered system to a desired state using a network control theory approach. And network control has previously been applied to understand how the brain's structural connectome can give rise to different neural activity states. So in this conception, a brain state is defined according to patterns of neural activity across all regions, and accessibility of each state depends on the structural connections between regions. So we can imagine representing different brain states along an energy landscape, where similar to a mountain landscape, states that are further away are more difficult to reach and require more control energy or input. Now, thinking about how we might apply this framework to a multi-layered system here, where we have, in particular, this is a duplex network, where we would give control input, which is represented by UT, to the input layer, which makes connections to an inner target layer, which we don't have direct access to. In this project, we are modeling functional connectivity and gene expression as two layers of a duplex network. So in the functional connectivity layer, edges between regions represent similarity in neural activity across time. Within the gene co-expression layer, edges between regions represent similarities in gene expression. And then we also have interlayer edges connecting each node to its replica node in the alternate layer. And those are weighted according to the ratio of neural activity to gene expression. So we envision by inputting control into one layer of the network, that we could drive it along its state trajectory and in turn also influence the state trajectory of the target layer with which it makes directed interlayer connections. Okay, so in order to apply this approach to the mouse brain, we are working with calcium imaging data from our collaborators in Mike Higley's lab at Yale. So to give you a brief overview of the experimental setup, the mice in infancy are given a sinus injection of a calcium indicator. Once they've reached adulthood, they undergo a calcium imaging experiment for a period of about 30 minutes, during which time they're just hanging out on this wheel and able to freely move. And they're able to record fluctuations in intracellular calcium, which are represented by fluctuations in fluorescent signaling, and that serves as our indirect measure of neuronal activity. So here I overlaid each of the Allen atlas cortical parcels onto the mouse cortex. This is in the axial plane, so you can see the mouse's right and left. The cerebellum would be here and olfactory bulbs here. And then over on the right-hand side, I've plotted time series traces from each of the available parcels. And this is over a period of about 30 minutes at 10 Hertz sampling rate. So 10 cycles or 10 time points per second. So in order to generate our functional layer, we wanted to capture dynamic changes in functional connectivity that happen over short time scales. So I generated functional matrices over each 3-second period from the whole 30-minute experiment. So each of these represents correlations in activity over 30 time points or 3 seconds. And that gives us about 570 matrices in total. So then to construct our duplex network, we have our function layer, which is each of those matrices that I showed you on the previous slide. And then we have our gene expression layer, which is generated using gene expression data from the Allen Institute. They have measured expression from about 16,000 genes in naive wild type mice. And so the matrix that you see here represents correlations in gene expression across all 16,000 genes between every pair of regions. So now using our duplex network construction, we can address the question, into which layer is it more energetically favorable for us to give control input if we wanted to drive the system to a desired state. So here are the results of our control analysis. Every point that you see represents one of the functional networks, which is paired with the gene expression network. So on the left-hand side, we gave input to the function layer to drive gene layer. And then on the right-hand side, we're looking at inputs to the gene expression layer to drive the functional layer. The modes represent input from every brain region. And so the takeaway here is that on average and across almost all modes, control energy is greater to drive gene expression from function, suggesting that it is more energetically favorable for us to give control input to the gene expression layer than the function layer in order to drive the system. Okay, now we have repeated the same analysis, except this time we're using human data. So here we used resting state functional MRI data from the Human Connectome Project, along with gene expression data from the Allen Institute, both of which were registered to the Schaefer 400 region parcellation, which you can see on the left there. And then again, we computed the control energy to drive system from either the function layer or the gene layer.
And what's interesting about this is that we see actually the opposite pattern as what we saw in the mouse. So in this case, we're seeing it's more costly for us to drive the system from the gene layer than the function layer. So to make it a little clearer, here's a side-by-side comparison of the human and the mouse. And the top row are edge weight distributions for the function layer and the gene layer. So another thing that stood out to me here is that in the human, we typically see this relatively normal distribution centered around 0 for both layers. Whereas in the mouse, we're seeing this very strongly left skewed distribution, particularly for the functional layer. And then in the control energy analysis, we're seeing that while for the human, it's more energetically favorable to drive gene from function. For the mouse, it is more energetically favorable to drive function from gene.
That's a quick question. So the mouse was running on a little wheel.
Yeah,
Turning cows. What was the human doing?
lying in the scanner quietly.
Not really.
So yeah, I'll come back to that shortly. So so in thinking about why we might see differences between species, it's useful for us to think about how in both species, functional connectivity and gene expression are biologically related. And this is a really nice paper where they looked at the relationship between functional connectivity and gene expression here in the human. So on the left-hand side, you can see they've colored several of the major human functional subnetworks. And then on the right, the little circles represent areas where they collected tissue to measure gene expression. So you can see overlap with each of those networks. And what they see generally is that genes that are implicated in encoding ion channels and neurotransmitter function tend to be strongly correlated with functional connectivity, which seems intuitive. And they also see that relationship between gene expression and connectivity in the mouse as well. And given the genetic similarity between humans and mice, I think it's probably not likely that the differences that we see with our approach are biologically driven. So clearly there's a lot more work for me to do, but some of the questions that I'm thinking about now are what accounts for the difference in duplex network control between mouse and human. Obviously, we have differences in temporal and spatial resolution with the different data types. We have differences in number of time points and brain regions. The two gene expression data sets are different as well. For the mouse, we have around 16,000 genes represented. For the human, we have around 10,000. It'll be important for us to confirm how much overlap is present. And coming back to your question, Julie, how similar are we considering different state spaces here when we're thinking about a mouse moving on a wheel versus a human resting in a scanner? How similar are those? So generally speaking, very interested in identifying conditions that contribute to how easily the duplex network can be controlled from either layer, which will help us better understand the behavior of the system in response to control input, and ultimately, hopefully, develop this into a tool that would provide meaningful insights and allow us to translate data or findings from mouse to human. So with that, I would like to thank my postdoc mentor, Danny, Pragya and Jenny, who helped out a lot with human data analysis, our collaborators in Mike Higley's lab, who did the calcium imaging experiments, as well as funding from the Michael J. Fox Foundation, and of course, the T32. Thank you, Anna Rose and Julie. And thanks everyone for your attention.
You- Yeah.
So.
Yeah, so it's not an addiction focused project per se, but it's a tool that I hope could be broadly useful for thinking about transitions between healthy and disordered brain states. So I think there's definitely potential relevance thinking about.
We talked about the reactivity states, ready states.
Yes.
Or you made some connections. I can swear.
Yeah, Greg.
You mentioned like.
Yeah.
About like something.
Yes. Yes, for the mouse.
For the mouse, yeah, I was thinking about the mouse, the go for data.
Yes, yeah, so the gene expression data are from in situs. And they are, so actually, one issue I had was those are aligned to an older common coordinate framework from the Allen Institute, CCFV2. And then we were working with CCFV3 for the functional data. So I ended up having to like lose a couple or a few regions because there was some mismatch just to get everything into the same space. But sorry, getting back to your question.
I was just thinking about specific cell types. There's a lot of functional cell types that work in agonist ways or come together. So I don't know if the buildings have something like that. The cracker states that like explains data or like good at costs from.
Yeah.
Cell types, I guess, with their one clarification, with their CMOS, you're not getting a single cell type, which is broad.
They do get single cell resolution, but this is just the mesoscopic signal that I'm looking at.
Okay.
But there is the possibility to look at single cell activity. Yeah, that's a good point.
Thank you so much.